Eh,
Hakikaten.
Good morning, afternoon, evening to all the invitees, speakers, and participants. Welcome to the second day of the international webinar on Fireflies from Bangalore, India. So yesterday we had the first day of the webinar, and very interesting papers were presented, and they dealt with the artificial light and living light, the biodiversity of bioluminescence, the emission spectra of bioluminescence, the light preferences or affinities by fireflies, the distribution of fireflies in Western Ghats, the firefly conservation in the IUCN Red List, the food choice and feeding experiments on the fireflies, and the impact of temperature on egg hatching, the role of ecotourism, and the study of habitat and behavior of firefly congregations in Kerala, South India. So, colleagues, this was some of the papers that were presented yesterday, and today we begin with a keynote from Dr. Sergio Hendricks. So, sir, are you there? Dr. Sergio Hendricks, Invertebrate Conservation Coordinator at the IUCN, Indiana. Sir, are you there? Dr. Sergio Hendricks? No, I think he's not there. But anyway, we have sent a message for him. We will see, he may join us soon. Then we will move on to the next speaker. Next speaker is Dr. Professor V.P. O'Neill from Wildlife Institute of India. Now, Professor O'Neill is a senior professor and scientist with the Wildlife Institute of India, Dehradun, Uttarakhand, and he is a member of the Royal Entomological Society, London. His specialization include ecology and systematic of insects, bioindicators, biodiversity, ecological monitoring in Himalayan protected areas. Dr. O'Neill was awarded UGC fellowship, and he has conducted many faunal studies, and he has also worked as the biologist under the FREP GHNP research project, and he has also participated in Nanda Devi ecological expedition. Now, these are very remote areas, very much difficult areas to work with, and he has also organized a number of international trainings and workshops, such as the International Mountain Conference 2022 from University in Innsbruck, Austria. Then he has also attended the mountain conference in several other countries. Then he has also participated on the Mountain Our Future Earth at the University of Highlands, Perth College Center for Mountain Studies, Perth, Australia. Sir, it's an immense pleasure that you have spared some time, your valuable time, for this webinar, and I request you to kindly make your presentation. Professor V.P.
Yeah, thank you, Dr. Chakravarti, for this introduction. I think so. It will better than you have to share my PPT from this side.
Okay, so we will share your PPT, is it?
Yeah,
okay,
please.
Okay.
It's more convenient for me.
Okay, yeah. So you can start. Meantime, in a minute or two, we will start sharing. Yeah. Please. Yes, sir.
Yeah, I can share. I'll wait. I think he's okay now.
Okay.
He's okay?
Okay, yeah. You want to share it?
Yes, comfortable?
Yes, it's comfortable.
Okay, fine. Yeah. Good morning, afternoon, good evening, everyone. Yeah, I'm V.P. O'Neill, already introduced by Professor Chakravarti. First of all, I am very thankful Dr. Chakravarti and his team for inviting me for joining this important conference on fireflies. So you already know this fireflies are declining from every part of the globe. But everybody's understanding the values of fireflies. And our study, we have conducted a very short study in Doon Valley. And the idea of the study was to assessment of fireflies, diversity and anthropogenic pressures in and distress of the fireflies population in Doon Valley, Western Himalaya. My student, Nidhi Rana, she is the PhD student working on fireflies of Doon Valley. She is a recipient of this second small grant of Robert Foundation for this study. So you know about fireflies. I don't want to discuss about this taxonomy. Just wait. Yeah. Yeah, there's known as the several names all over the world, but in areas of Uttarakhand, we are told is in Hindi as Zugnu. And there's a lot of folklores, small poetries in the school level, some Jugnu. And that's families. Lantharidae, you know about it, short body beetles, commonly called as fireflies, because of this conspicuous use of bioluminescence during twinkling that's attack to the midst of the prey. But the documentation of the fireflies is not accurate. In the worldwide information available is 2,200 species. But in the case of Indian status of fireflies, the data are deficient in India. Somebody is saying that 55 species, 50 species, 74 species, but clear information of region-wise of fireflies has not been documented so far. So some of this insect, the group of insect we are having the Phonobritis India, like Scarabeetie, tiger beetles, other beetles, but not available on this information of fireflies. And therefore, we understood that is a need of the study of fireflies, and we developed the proposals and assessing the diversity of fireflies in Doon Valley. Global status 2,200, and nearly 50 species are known to be India as report of Zoological Survey of India, because only the authentic institute of a country who is giving this documentation. They're having 4 subfamilies. Lantharin is a common one, and about 8 to 10 common genera has been documented, and information is available here. So the life cycle, I don't want to discuss about it. Everybody expert of fireflies, they know it is eggs and then larvae phase in the longest phase in the firefly lives. They live in the soil, leaf litters and predictions. The cause of decline of fireflies are also the leaf litter situation, because of this forests deforestation, pesticide use, and leaf litters impacting. That is also very crucial problems of the population of fireflies, and it's adult show sexual dimorphisms, and it takes mating to 3-4 weeks. Pupae after 1 and 2 years are converted into pupae, and every stage of fireflies life cycle possesses the ability having the bioluminescence. So definitely declined fireflies population. A lot of articles all over the globe, in India also. Some of the scientists are working, and they are very serious, and they have published the documentation of fireflies declines. But in Indian, taxonomic studies of fireflies have been not done very seriously. Thus, our project, we are having the objective here to assess the diversity and factors influence to the firefly population in different habitat types of Doon Valley. Doon Valley is a cup-shaped structure surrounded by the Shivalik as well as the lesser Himalayan forested area, and escalation of fireflies conservation awareness among communities through outreach programs. How you will communicate to the society about the conservation of fireflies. Everybody is talking right now that their fireflies are declining, their sighting is very, very less earlier than we. But what is the cause, and how can you communicate this information to the community? This was our idea. This is our area study we have designed here in Uttarakhand, and we have done is the 4 forest ranges of Doon Valley. Doon Valley is surrounded by this agricultural lands as well as the sal forest, and upper region of Doon Valley is the lesser Himalayas, the mixed forest, as well as the Shivalik. So only we have just documented the information in the 4 forest ranges. That is each range having divided into 4 different habitats: forest land covers, water land covers, agriculture and land covers, and urban land covers. These are the 4 parameters and 4 divisions of each range we have divided, and within that ranges we have done this 100 to 100 meters plot grid, and within that 100 to 100 meter plot, 25 to 25 meters subplot within that plot for documentation of the vegetation structures. So the every plot, every forest types, and every habitat has been designed very systematically in all 4 ranges. So this is the landscape and habitat types in forest area is the complete Sal forest of Doon Valley, surrounded and part of the Rajaji National Park. This is the urban area of Doon Valley. Riverine areas, there are three rivers in surrounded by Doon valleys, and this having heavy pressures of silt and other extraction of those areas, and the agricultural lands in the Doon Valley also. Earlier, you know about Doon was famous for the special kind of rice, the basmati rice. Now, the whole agricultural farms has been converted into habitations, urbanization. So this is one of the impact also. So these are the whole 4 ranges and 4 areas of habitat we have documented. Yes. So this was the designing of our study, the 100-meter plot. Within 100-meter plots, 25-meter plots have been distributed as coordinate samplings for this, each format for each land covers has been done in all 4 ranges. Within 4 ranges, all 4 habitats. So all the sampling plots were marked with color tabs. Random samplings was done within the plots, and inventory of vegetation was made within the coordinates. So this plot was completely marked, and we have done the repeat documentation of last 2 years. And this is the going over the 3rd years. So we are having the consequent data of 3 years of each habitat and each plot. So different ecological variables such as temperature, humidity, canopy covers, wind speeds, light intensity was also documented using appropriate instruments. So this is the area and this is the methodology which we have designed for documentation of fireflies of Doon Valley. So well-tested methods of collection of fireflies, sweep net and pickings in each area. So these are area that we have done is samplings, conversions with the locals community where they have seen the fireflies. Somebody villagers, they are saying, yes, we have seen the fireflies. Here last year is a very large number, but this year is not. On the basis of the local, I can in the case say it's the secondary informations and forest areas. So that is the regular exercise we have done in last 3 years. So this year exercise is going on. These are our results. So all 4 ranges we have documented is Thano range 5 species, and Barcoat range 3 species, Asarudi range 5 species, and Malan range is 3 species. Some of the species is common and some is restricted on areas. So for after 3 years, then we can get a very concrete information why particular species is available in one range because of that habitations and the kind of regions. So we can designate some indicator species. So on the basis of our complete set of data, then we can identify the particular species in particular area can become the indicators of habitat shares on the basis of those collection and those documentation ways. So actually the common Zenda is Condita, Lambideras, and Asymptoticata. So this is we have documented 6 species or 3 genera. The species can be distinguished based on this morphological and gentile characters. We have done the DNA barcoding of those species. This is on almost everything, all results are available, and I think so it will take 2 weeks more to confirm those species, but we have done the DNA of sequence of all 6 species. So this is some of the distinguishing characters of species level as well as genera level. It is known, it everybody knows it. The main features of terminals, tergites of the case of Abscondita, Lamperigera is having this pronotum is different colors and size of pronotum. You can easily identify this genus and species, and Asymptoticata is already having this features of light organs. So this is the threats of observing in this area. What is the cause of the threats? Definitely habitat loss. I already discussed it. Climate change of various regions. This year also, there's winters very, very vast in Doon Valley. Monsoon is, you know, that in Uttarakhand and Himachal and western part of Himalayas is calamities of this year's due to the monsoon. So this is one of the region of this climate change. Pathogens, agricultural practices because of that intensive practices and using of pesticides and fertilizers, invasive species and loss of plant communities. So this, I think, so which is I'm observing that people are mostly using this cash crops and exotic species of plants. So it is maybe cause of decline of fireflies, and the studies needed for this intensive areas. So the fragmentation, you can see this is Sal forest of Rajaji is very close, and that it is a deforestations and kind of habitat degradations in areas, and it is cause of the decline of forest fires and light pollutions. We are now using this high bright LED lights. Earlier, we are using this different bulbs using yellowish lights on street as well as our house and our porches kind of things. Now the light structure is changed. Aeroplane, airport areas, bus stands, community center, roadside, every area is having the LED and very bright lights. It is one of the cause of decline, and light pollution is declining for the population of the fireflies. Construction urbanization is worst affected. The west area of Doon Valley having this bypass, national highways, bridges, and construction material as well as the reverse end system, they are degrading it. So this is one of the cause of the decline of fireflies. Not only the firefly, other species of insect also, and animals and wildlife. So this is the common features and common pressures of everyone in biodiversity. So use of pesticide fertilizers, we are communicating it. Everybody's government is having this saying that is go for this biocompost and organic farmings in Himalayas. Some of the states like Himachal Pradesh, Sikkim, they having declared the state as a organic areas, and even an organic board in Uttarakhand also they are giving this instruction to the farmers: don't use the pesticide and fertilizer. But still, large number of farmers they are using the pesticides, agricultural fields. This practice is on. I think you have collected some of the bottles in the agricultural fields also. So this is one of the cause of this decline of fireflies. But we have done our level best to communicate this information to the common man. And we have, it's part of our project of second objective that this citizen science approach for this conservation. So awareness events, we have done it. And what we have done, we have countrywide surveys have done. And on the day of World Firefly Day, 3rd and 4th July, we have done in 2021, 2022, and this year also. We have designed a small format on mobile, and that information we have circulated all over the country, all forest officers of the country, because of through our directors, Wildlife Institute of India, we having our contact and association with the forest department of our country. And for this survey, like we have generated this, circulated through this website of Wildlife Institute of India, Facebook, WhatsApp groups, all social medias, and survey provided to the occurrence of data file. It was very easily defined. It is the name, email address, data and time of sightings, number of fireflies they observe it. If they have seen no firefly, can zero it. It is zero is the result also. It means there's no fireflies. And our suggestion, if the things is available on the photographs is available, then we can say that upload the photographs. So this information we have circulated last 3 years to every state, every forest officials, frontline staff, forest guard, range officer, DFO, PCCF, and NGOs, university students. But we got this information in 2021. Only 71 participants contributed of 14 states. And it is for 2 days. We already given instruction in every evening hour, 7 to 8, how many fireflies. No need of any species identification, just presence of fireflies. If not, say no, zero. So we'll get the information that fireflies is available or not. In 2021, we have represented and we have got 73 participants in 14 states. And after submission of their information, we analyze all the data set, and then we have circulated and given this certificate from our directors for this participations and their thanks letter. So we have just published a paper on that firefly surveys, adopting citizen science approach to record the status of flashing beetles. In 2022, that early we have got the information from 19 states, and 190 participants submitted their information. So we're having the 2 years data. And this year, we got only very limited numbers, and that is only 14 species, 14 states represented, and number of data is, I think, so 100 something available. So it was less this year. But we have done our level very best to communicate each and every person dealing with biodiversity, dealing with wildlife science, biological students, to participate in that way, to kind of cover this. So through media coverage, already is lot of local papers and print media as well as our electronic medias. Nidhi has I already given this information to the local radio that can say the FM radios of all our programs. On this date, Fireflies Day, we are circulating this information. You are requested to give the informations and conserve fireflies. We have done this exercise and large number of school workshops, community level workshop, the school level workshop has been conducted it because it is a part of that project. And since last 3 years, we are doing this approach and communicating to everyone. So this was a brief information about how we are doing this exercise, and we are having only 6 species documented. Still, our program is to evaluate those information to the part of Western Himalayas. So we are very thankful to their directors, Uttarakhand Forest Department. They communicate and they give the permission to do study also. Rafford Foundation is financing, giving assistance for this exercise. Second is the second grant we have received. And definitely Dr. Leslie, she give the ideas. He already give the identification and support and all those technical support for our team. And basically, this Nidhi is contacting us most of the times, and this was the information about the fireflies of the status of Doon Valley, which we have done since last 3 years. So this is the about from my side. Yeah, thank you very much.
Yeah, sir, thank you so much for informing us about the activities at the WII. Which is a western part of the
No.
Yeah, any questions to the speaker, please? Yeah, sir, I have one small question. When you have received the information about the occurrence of fireflies from different parts of India, and the number seems to be varying from say 70 to 190 or 40 to 190. Yeah, have they indicated the species or the general?
No, not at all. We already tried to contact JSI and other scientists. JSI is saying there are 50 species, and yesterday there's some of the speakers, they say there are roughly 4 species documented, but the details, which species from which region, is not received any information. And we have not done a very systematic studies on firefly, very unfortunate, but we are trying and we'll collaborate with you also. Yes. So we can plan a small project, joint project. We can take the help of Leslie and Dr. Leslie also.
Sure.
So we can do a documentation. We are like a pollinators, which group I'm also working. So we are having a joint approach and joint scientists of different organizations and working on the status of pollinators of the entire country.
Okay.
Other group I'm doing out in the entire information of tiger beetles of the country.
Okay.
So we can plan it. There's a complete information of fireflies in the country.
Sure, sir, and definitely we will have the guidance.
So we can you can submit a joint proposal, and some area has been divided. Some of person working on Northeast can do the study of the Northeast. They can do it Western Ghats and Central India, and I can plan for this Western part of the country. And definitely then we'll be got some information.
Yeah, so we are very happy to know that there is a strong group on fireflies working under your guidance in WII, the western part of India, and definitely there will be other teams working on fireflies in different zones of our country, so that we can pool all these people, have good, skilled, trained taxonomies on fireflies, at least for our identification. So thank you very much, Professor O'Neill, for enlightening us on the fireflies and the extension activities which you have taken for their documentation and conservation. Respected participants, we will move on to the next speaker, Dr. Dhamika Vachikon from University of Ruhuna, Sri Lanka. Now, is a lecturer at the Department of Zoology, Faculty of Science, University of Ruhuna, Sri Lanka. And he has put in over 15 years of extensive experience in identification and taxonomy of Southeast Asian fireflies. He is also interested in birds, animal physiology, and wildlife photography. Dr. Chandana is a member of the Firefly ICN Specialist Group and member of IUCN Firefly Program funding. He was very kind enough when requested to share his work and experience on Firefly in this webinar. Dr. Chandana, please.
Okay, can you hear me clearly?
Yes, we can hear you.
I think you can see my screen sharing also. No?
Yes.
Okay.
Yes.
Okay. I am Dhamika from Sri Lanka. Actually, nice to hear sharing my information with this kind of platform, because Dr. Valentine also here, because she helped us numerous ways for the emerging this kind of fireflies in Sri Lanka. So, and I especially thank Professor Rakesh Chakravarti and his team for organizing this kind of webinar in India. I think it is a second time, or anyway. So, thank you very much for inviting me and organizing this kind of webinar, because it is a good platform for sharing information between actually the firefly experts, and especially this is important for young students and researchers who are willing to study on fireflies. No? Actually, so my topic is overview of fireflies in Sri Lanka. Just I selected this topic because we are in the neighboring countries. I think that just the information of fireflies in Sri Lanka will be important for Indian scientists and researchers also. So, okay, this is my content. Just brief information about the species diversity of fireflies. Just I'm going to talk about fireflies we confirm identified since 2010, because then their habitat diversity and some human-induced threats we identified so far within the country, and their conservation recommendations. Okay, this is the location of Sri Lanka, I think, because it's close to the Indian subcontinent. Since ancient times, we have good relationship, because we're sharing many more aspects regarding the culture, religious, and everything. And we are in the same tropical region and the same biodiversity hotspot. No? Therefore, I think we are sharing some biodiversity also in some extent. So this is the summary of the fireflies in Sri Lanka. So far, we identified so many subfamily Luciolinae fireflies, and then moderate number of Lampyrini fireflies, and these number of Autotretinoid fireflies. Just I'm going to show you some actually these fireflies so far we identified, just their distribution and some kind of the other important features we encountered during our field surveys. Here, sorry. Okay, it's a subfamily Luciolinae, the first time perplexa. I think India is actually this species is also common in India, because actually we encountered this species throughout the country here. The country represents 9 provinces, and I think this species we encountered in almost all regions, and especially during the actually the not any seasonal, because we encountered this species throughout the year. Actually, this is one of common firefly in Sri Lanka. We can see more abundantly throughout the country. Then just moving to the next one is Abscondita pramalina. Actually, this is a pale color one. It is distinct and restricted only some coastal areas and some highland areas of the country. So they are actually, we observe these species. They more common in high elevations. Okay, so this is a pale yellow firefly and with dark mesocuculum. And then moving to the next firefly species is Symmetricata humeralis. It is also common in India. I think this species also we encountered all throughout the country here. This is an year-round variation we can see this species. Then moving to the next one, Impressa, it is somewhat similar with the previous one, but there are some distinct features in thorax here. So this is so far we encountered this in only central and southern parts of the Sri Lanka. And moving next one, the Curdos costipenis. This species first recorded in 2010. Actually, it is only one species were recorded under this genus. With the help of expertise in other countries, these taxonomists, they help us to identify this species. They are very common in some regions in Sri Lanka, especially. This species represent both dry climatic conditions, climatic areas, and also the wet climatic areas. This in the northwestern province represent the both dry and arid zones, but they are common. And also these areas represent the wet zone of Sri Lanka. So we encounter this species from these regions also. Then Extricans, it is also one of the original described firefly species from Sri Lanka by European scientists in 18th century. And as you can see here, it's a distinct black apical areas of elytra and the square shape black mark on a pronotum here. Their distribution also restricted in actually the southern part and the coastal zone and some other mid-country areas in Sri Lanka. Then Nicola Rai, I think it was this species we recorded in 2022. And the special thing is actually the special thank goes my students, Mr. Dishan, because he encountered the specimen, he collected the specimens here. They are actually we recorded the species only from southern province so far. It's an intermediate forest. And important finding is we found there are females also. It's a flightless female. Then next one, Luciola horn, it is also one of rare species we found in Sri Lanka just after first recorded in 2010. Unfortunately, we couldn't find any specimen of this species so far because we first recorded species from Uva province of Sri Lanka. It's a kind of a very rare firefly. Then next one, the Candace. It is you can see here it's a bright orange color, and we encountered only female. We try to find their males, but unfortunately we couldn't find their males yet. We are still investigating their sites. It's also definitely this species also originally described by using female characters. So then we recorded this only the southern part of the Sri Lanka. It's a kind of a large firefly. And Sclerotia substriata, actually this species initially identified, originally described as Luciola cingulata, but now with the recent taxonomic revision, the genus and species goes to the new genus and the combination Sclerotia saturata here. This special thing is so far we recorded this species from 3 provinces of Sri Lanka, the southern Sabaragamuwa province and Uva province, but especially freshwater associated habitats, a kind of good indication we encountered. So we are investigating the immature stage also. Then Pygoluciola, this is a kind of this genus we recorded recently for the first time in Sri Lanka with the help of Dr. Valentine. And actually this is our Pygoluciola species somewhat distinct than the other Pygoluciola recorded in South Asia, with having this pale, that's a yellow, bright yellow dorsum, and the more white, actually the lateral white elytra. Then this is the second one, Pygoluciola species 2. Actually, this is a kind of a pink firefly. It's a pink pronotum, and there are pink some apical marks on the elytra. This is also found. This both Pygoluciola species we found from intermediate zone of Sri Lanka, especially in the southern part of Sri Lanka. Then I'm moving to the next subfamily, Lampyrenae. So this is a common in India also, Lampycera tenebrosa. So we encountered this species in almost all regions of the country. Their distribution is common, and we observe this species throughout the year. In Diaphanous luteins, it is also one of original described firefly species from Sri Lanka. So we encountered from 3 provinces so far, and they are immature stage, especially females and larval forms also. Then Diaphanous vitrifera, it's another one of rare firefly we encountered, only so far recorded from the Uva province. With the water associated habitats, it's a distinct pink areolar area on the pronotum. It's actually the transparent and the very beautiful firefly species. Then I'm moving to the third subfamily, Ototretinae. So this is Hematilia bilinea. So this species is endemic to Sri Lanka because when reviewing the empirical literature, we found that there are 3 species of genus Hematilia recorded from Sri Lanka. This because Hematilia, the genus Hematilia is endemic to Sri Lanka. So far, we encountered actually with our recent studies, we encountered 1 species. This is a kind of we observed they active both diurnal and also the nocturnal. They show the bioluminescence activity, and we found some female stage also. It's a kind of beautiful firefly. Then Eugesis palpator, it is another endemic firefly from Sri Lanka. It is also, they have this kind of some bioluminescent organs. Actually, it's not the light organ, it is kind of bioluminescent. We observed their bioluminescent activity at night, and they also active at both daytime and also the nighttime. Then I'm moving to the last one, the Stenochladis species. It is also have bioluminescent firefly. It's kind of belongs to the Ototretinae, and they also be found active in day and night times. Just this is the species so far we identified some with our recent studies, and then I'm moving to the next part, the just information about their habitat diversity. We encountered fireflies from terrestrial grassland habitats and the paddy fields and freshwater associated lands and forest. And recently we encountered some species even in coastal habitats also. Then just threats I'm going to, because I think in the previous talk, actually you aware about these threats and conservation, some kind of threats, and this is the threats that are declared by the IUCN Firefly Specialist Group. As in Sri Lanka, we also have some experience on these kind of similar threats. So the habitat destruction, light pollution, and use of pesticides. Habitat destruction, this is I extracted this picture from the IUCN Firefly groups. It's it has explained how to change the natural habitats, and this kind of mangrove habitats. They are specific habitats for somehow the high diversity of fireflies in some areas. So therefore, when they transformed into a new habitat, that means artificial habitat, their original population will vanish. Then it's one of examples. This species from Sri Lanka, Diaphanous vitrifera. Actually, this is their original habitat when we encountered them in 2010 in the Ua province. But now this actually it is a water-associated habitat, but now it has transformed into the hydropower plant. Now they all have actually this habitat now vanished. So therefore, this population we couldn't find. Actually, this species we couldn't find from this habitat. So one of the good examples from Sri Lanka. Then the development activities, the development of accessories, that means infrastructures for the natural habitats. So this kind of concrete roads, they are severely affect the life cycle of the fireflies, especially they are in mature stage. So then moving to the natural water canals. Actually, this is a very common recent in Sri Lanka, because it's a kind of new trend that's involved by agricultural system in Sri Lanka. So the despite use of this kind of natural water canals, with the implementation of new projects, so they have developed this canal using the concrete. So it is a kind of severely affected by the firefly life cycle, because it is a natural habitat is important for their survival, especially in mature stage. So but now actually with this condition, they are in mature stage, cannot move, and their feeding is also not available, especially snakes and slugs and other mollusks. So therefore, this is a kind of a severe threat for the firefly's survival in Sri Lanka. Then, so just for information, I extracted this photographs from the internet. So this is the Sclerotia aquatilis, their life cycle. This is species not in Sri Lanka, but just I use this diagram for the understanding, because this kind of developments, water canals severely affect this actually the water associated or the aquatic or semi-aquatic firefly species, because their food source are not available with this artificial condition, and water plants and the water grasses we cannot see in this kind of, because water is moving very fastly. So that is a problem, and it's the surrounding areas. Hello, can you see? Hello, can you hear me?
Yeah,
okay,
yeah.
okay,
You can
okay.
continue. You can.
Okay. Thank you very much. So then, this kind of associating habitat, associating soils, it is important for the survival of the especially aquatic and semi-aquatic fireflies. How nowadays we are facing this kind of threats, actually with the development of water canals using the artificial substrates. As I mentioned earlier, this species we encountered from water-associated habitat, but now we actually we have data. So to illustrate that, now their population is decreased. So in few numbers, because in their habitat we encountered previously, that means in 10 years ago, sometime in 2010, actually we encountered species so many in water associated habitat, but now we couldn't find them in more common. And water and soil pollution is a common threats and the common problem in all over the world. No, so this is a another threat for the fireflies life cycle. And pesticide, insecticide, and molluscide is also another threat because you know the Sri Lanka has an agricultural economy, so therefore so many pesticides, insecticides, and actually the molluscide they use for their agricultural sites is a kind of severe threat for the fireflies and their food items, molluscs. That is a major threat we are facing in the country. And the light pollution is a common problem and throughout the world. For that's a threat. They are the behavior of fireflies because as you know they always prefer to dark habitats and therefore they always move away from their artificial habitats, artificial light source. Okay, conservation recommendations. Actually, this are our recommendations. You know, there are so many experts in this field, but just I am going to point out what are the recommendation of us. Actually, the protecting of their natural habitats and the reducing artificial developments of water canal systems. That means natural water canals. It is a major problem. And the minimizing the use of pesticides, we have some alternatives, but if we can promote the farmers to use this kind of artificial, that means alternatives, it is important. And the avoided light pollution, restoring their habitats and the society awareness. Actually, within this past years, we always spend more time for awareness, that means using media and the using awareness of the society. People use workshops and some programs and paper articles, so on. So I think this is one of a good solution for the reduce the water pollution. No, it's a major problem is the street lamps nearby some sensitive habitats of the fireflies life cycle. That is a problem. So, but in developed countries, they use this kind of sensory systems. It is a kind of a good alternative for the, and this kind of street lamps that are focused only the road, not the surrounding habitat. These are some alternatives. Just okay, that's all. This is our research team. Actually, the Professor Hemanta and Mr. Dishan, and actually, I greatly appreciate Dr. Valentine also. She helped us to identify every specimens, especially Luciolini. Okay, thank you very much for giving me this opportunity. If you have any questions, now you can ask.
Thank you very much, Chandana, for you dealt with the important observation and the way forward. Dr. Chandana, there is one question from Nitin KS. Recently, the government of Sri Lanka banned the import of fertilizers and pesticides. So do you think it augurs well for the fireflies and other biodiversity elements in Sri Lanka?
Yes, I think it is temporarily banned by previous government, but nowadays it is again. I think they are importing. That is a problem. But because they promote the organic farming, but I think it is okay if we can implement.
Yeah,
this is safe for the firefly life cycle.
Yeah. There is one more question from Ishwari, Dr. Ishwari. You mentioned that UGSS lacks evident photic organ, but still present the bioluminescence.
Yes, we just observed because bioluminescence at night, and they are also active in daytime. That's so therefore because they have well-developed antenna and for the communication. It's a common feature of the, I think, autotread name groups.
Yeah. There is one more question from Dr. Ved Prakash Singh. Can we get some knowledge upon introduction of fireflies from one area to another, especially to areas where fireflies are vanishing?
Yes, because I think we studied some areas with where 10 years ago. We observed that their fireflies are very common, but nowadays we observe their population is very limited in the areas. So this kind of information and the observation we can encounter.
Yeah. So that's good. Any more questions from participants? If you have any questions, so we can just contact directly, or you can just mail us or contact us, and we will forward the questions to Professor Chandana. Dr. Chandana, thank you very much, and we'll be in touch with you because we're also working on Firefly, especially on congregations and the impact of traffic and traffic light during night on the Firefly congregations.
Okay.
Thank you so much once again.
Okay, thank you very much.
Yes. Ladies and gentlemen, we move on to the next presentation by Sergio Enriques. Professor Sergio Enriques is the Invertebrate Conservation Coordinator at the Global Center for Species Survival, Indianapolis Zoo, and is the chair of the Conservation, Sustainability, and Land Management section at the Indiana Academy of Sciences. Professor Sergio has more than 20 years of experience mobilizing resources to promote invertebrate conservation, looking for lost species, and in summer, he can mostly be found under Indiana's dark skies, studying our state fireflies. He regularly works with a global network of experts, including IUCN specialist groups and the Invertebrate Conservation Committee, to tackle unstudied threats such as illegal trade and also the light pollution. The latest technological tools to reverse the ongoing decline of the most diverse group of organisms on planet Earth, the invertebrates. Now, Professor Enriques is not physically presenting the presentation because of his preoccupations. So we will just play his video, which he was kind enough to send us now. Professor Sergio Enriques, please.
Hello, thank you so much for inviting me to be a part of this meeting. I'm sorry that I can't be with you live, but I'm looking forward to listen and learning from all your presentations and work together towards accelerating conservation action for fireflies, specifically in India, but also in my professional experience, or my professional goal is to do it at a global scale. So without further ado, let's do a small introduction. So I'm Sergio Enriques. I'm originally Portuguese, and when I was back home just a few months ago, I saw this beautiful female of Lamparisa, and even though I'm now in oceans, apart from my home country, I'm now in Indiana, in the Midwest, I still do see blue ghost fireflies, which belong to the same group of fireflies, and they're actually quite similar to the ones I know from my childhood. And on the corner of this map, you will see a picture of fireflies from India that I thought would be nice to share with you all. I chose the blue ghost firefly because not only has a connection to my home country, but it also has a connection to the fireflies in India, because much like some of the firefly festivals in India, the blue ghost attracts tourism, is a species of interest and beauty for a lot of people who gather here in summer nights. And I think it's a good entry point to see where fireflies around the world come together as being, of course, an object of attraction, but also a source of responsibility. We are responsible to try to support this tourism industry to be sustainable. Tourism and visitation are often connected. People come to cities and places for a number of reasons. I work in a specific center, the Global Center for Species Survival, which is based at Indianapolis Zoo. So we work a lot with visitors, and I understand that there's potential there to educate and to spread conservation messages to the public that comes and sees the beauty of animals either in nature or in captivity. The Global Center for Species Survival is a partnership between the Indianapolis Zoo and the IUCN, the International Union for the Conservation of Nature, and the Species Survival Commission, specifically within IUCN. The Global Center hires a number of people. There's pictures of our team in the bottom of the slide. That's Bill Street, our director, our mammal coordinator. We have a plant and fungi coordinator, a marine coordinator, a bird coordinator, freshwater coordinator. I am the invertebrate conservation coordinator. So it falls on me to help the specialist groups under the Species Survival Commission to accelerate action to protect and reverse threat on extinction risk of all invertebrates, but fireflies in particular. I mentioned fireflies specifically because Indiana is a special state here in the US, because the state insect of Indiana is the firefly. And actually the picture that I'm sharing with you now is the school children that led that initiative to have a firefly as a state insect, and this is the day that the governor accepted or signed that agreement that made it official. The state insect of Indiana is the Say's firefly, or Paracnomina angulata, and that's a picture of it just next to me. And as you can see from the red list assessment of that species, population is thought to be decreasing. But our knowledge is so poor about this species that even in the state where this species is the official state insect, there is no record. In the map, you can see that line in red, that's the state of Indiana. None of the blue dots are within it. That means that no record, no observation was taken into account when listing this species or when red listing it and assessing it. We just don't have information about where it exists in Indiana, let alone how its numbers are doing. So as I hear colleagues from India saying that we don't know enough, we don't have data, we need taxonomical experts, we need more geographical distribution, I understand that. That is the issue also in the United States. That's the issue also in Europe, where I come from. And I'm guessing it's the issue for a lot of fireflies around the world, especially Africa, but anywhere. And the same for a number of other insects and even other inverts. However, these animals are so charismatic that there's a potential for fireflies to become flagships, conservation flagships for other species or umbrella species that will attract conservation efforts, and that in turn will protect all the animals in that habitat. The pictures below are some that you might know. These are the pictures that, to me, at least rose my awareness about firefly diversity in India. These pictures went around the world. They were widely shared, and they are, I can see why, they're remarkably beautiful. And they do alert people to, this is something that we would like to protect. This is a legacy we would like everyone to enjoy, and we would like our children and generations to come to enjoy as well. And here is how I think we can do it, how we can protect these species. So the IUCN has put forward what they call a conservation cycle. And usually the conservation cycle has three main components. And one of the ones that's most famous to people is perhaps the assess component, and that is the red list, as well as many other tools, but the red list being perhaps the most well-known. The red list, as I mentioned in other presentations, places or assesses species extinction risk from, of course, extinct, the animal is no longer fine, or plant or fungi, all the way to least concern, meaning that it's not an immediate priority to the best of our knowledge. And there's, of course, three threatened categories and other things in the middle, but that's the assessed category in a nutshell. What I would like to emphasize is that that's a cycle, so there's other components. One of those being planning, and that's, of course, led or has a lot of good guidelines by the Conservation Planning Specialist Group, which has training and has a number of tools online that I recommend you to look. And with that assessment information, you can do very good conservation plans, action plans. But none of that will really matter in the end if we don't act. And that's really what I want to emphasize or focus in this presentation, is that action is, in a way, one of the most important components of the cycle, and one that I think we should encourage or support more. I also want to highlight that this is a cycle, meaning it doesn't have to begin in assess. It can easily begin in act, it can begin in plan, or it can begin in assess, of course, but it's a cycle. It's meant to be repeated, and it's meant to be begun wherever opportunities lie. An opportunity to accelerate this cycle is, for example, doing assess to plan. There's a really good YouTube video on this. If you just write assess to plan on YouTube, you'll come across this video from the IUCN Conservation Planning Specialist Group, CPSG, which will explain more. But in a nutshell, the assess to plan takes advantage of those experts that are together doing the red list assessments. So already know the species risks, already know the threats, already know its biology. And once they're together and they're assessing a number of species, let's say red listing all fireflies from India, as they do that, in that information, in that working group, there's already capacity to see this is a threat for half of the species and this is the main threat for the other half. So as you're assessing, you already have the knowledge in place to try and think about what plans could be in place that mitigate or that help most species or a number of species to be better, to basically have a lesser number of decline, or to improve its population status or its numbers or its range. So there's an opportunity as we assess threats to also plan on how to mitigate for them. I think it can be intuitive how one can begin by assessing the red list. And of course, then you'll come into issues about what's a true range of the species and others, but it's a bit harder to convince people to begin with planning when we don't know the extinction risk. But if you are locally based in a region, in a reserve, or if there is no protected area, you already know that that's a threat. Habitat loss is already likely a threat for fireflies and most other species in your region, as is light pollution, other forms of pollution like chemical pollution, reduction in water levels or water quality. So often we don't know where the species exists, how long it lives, what it feeds on, but we have an understanding of the problem. And there is a tool that I'd like to share, which is adaptive management. And if you have an understanding of what the problem is, what problem might be for those species, then you're in a good position to go to a design stage where you can think, habitat loss is an issue. How can we design so that we can improve or recover that habitat? And questions might arise, maybe you're not recovering their habitat, the right one, maybe not recovering it the right way, but there's an opportunity there to do an adaptive management sort of experience where you can, let's say, take a plot of land that you want to recover. You don't have to imply or implement the same amount increase? Have you been seeing more of that in that place? So really, as you do an experimental design of mitigation of threats or recovery of habitat, to that extent, often we don't know the impacts it will have. But if we wait until we have that knowledge, it might be too late and we might lose too much. So my proposal here is that sometimes it is better to have a sort of a design and just implement action on the ground based on the knowledge we have from other places. We already know habitat loss is a threat. Recovering habitat could and should be a priority. And of course, maybe it's not done in the best way, but if you implement it and then you monitor, which is the cycle in the bottom, if you monitor the differences, you can then evaluate what worked and what didn't. And if it didn't work, you can adjust it. And then you assess the problems, what other problems came across, and you complete and you continue that cycle over and over again. And that's, in a nutshell, adaptive management. And that's, I would say, is a good way to do action in a manner that's effective and that puts forward solutions. Because if we wait to have all the information, it might be too late to save some of the most remarkable species on the planet. I have a few examples to share of this. We have mentioned often that light pollution is an issue specifically for fireflies. And when we try to increase security or when we allow for visibility in roads, like in the picture I'm sharing, you basically might be creating a barrier to these animals, which for us, of course, an open site with very distinct poles at large distances, but for a firefly, that has a high level of visual acuity in the dark and is reliant on that visual acuity to communicate and to mate. That's a barrier. And so one of the ways to, of course, prevent this and see the impact is just reduce light pollution. If there's ways to do it safely and do it in a way that's sustainable and that is supported, of course, by local stakeholders, reducing light pollution might be a good way to just reduce barriers. One of the biggest threats people will mention that seems unsurmountable is climate change. Climate change is not unsurmountable. People can do everyday actions to try and minimize their impact, to talk to policymakers for them to enact change and, of course, have an impact on the companies that, in fact, have the biggest, highest impact on carbon production. But when we think about conservation action on the ground, something people can do is climate change resilience. And that often comes with knowing that as climate changes, likely species in the northern hemisphere will move or will need to move north. Their range will have to move because southern parts of their range will become unsuitable, while northern parts of their range become opportunities for them to move towards. But they can only do that if there's connectivity. So there's an opportunity to create connectivity for fireflies. And as people here might be interested or working in captive rearing, captive breeding of these animals or doing lab studies, there's an opportunity there to have these animals in captivity, boost their numbers so that we can accelerate that movement, so that we can place northern populations at a stronger population number, boost them in effect, so that they can endure that move. They can basically lose individuals to move north and still remain strong while connecting them. So I know this is complex and I don't want to go into details, but these are the kind of ideas that I would like to share. I mentioned habitat lost a few slides ago, and it's quite easy to see in the picture that I'm sharing here, who was, by the way, picture taken here in Indiana, that sometimes is very easy to see what is missing. There's forest was taken. We need to replant that forest and accelerate that process if we can to recover that habitat. It's not always so clear, but like I said, adaptive management might give you some opportunities, and it's a tool worth considering. If you don't know what habitat would be more advantageous, would support more biodiversity, but in land swaths that are relatively big, there's a chance to divide it in plots and enact different types of action. And often, to be honest, locally people will know what is the best way to accelerate plant growth of different species. So that knowledge sometimes already exists. It's just a matter of gathering the right knowledge to accelerate or promote habitat recovery when there's been habitat loss. One of the other threats that's often mentioned is insecticides. People will talk about pesticides or herbicides, but really insecticides is what might impact fireflies the most because they're already targeted at beetle species, and fireflies are beetles. So it's likely that the same chemicals that will kill a pest beetle will also kill native species of fireflies that, of course, are not a pest to crops. But crop production is an important side of human development. We need to produce food at more affordable prices and higher quantity to feed more people. But if we want to do it in a sustainable way that supports both populations and wildlife, there's an opportunity here as well to do adaptive management and working with farmers to see if you don't use this herbicide, this pesticide, this insecticide, but use this other instead, would that benefit fireflies? If instead of having the runoff from the farmland going straight into the watershed or basically into a creek nearby, are there mechanisms we can do that kind of isolate that small patch of farm? And so there's opportunities here to try a few methods, try a few actions and interventions and see if that will impact firefly or other species biodiversity in numbers. The examples I shared, I think, are more or less intuitive, but none of them will really work if you don't have the support of local stakeholders, those who actually live in these places. And of course, you can't really do it without funding, without financial support. There's not much that I can say about how to promote local stakeholder engagement. Apart from saying that is really important, no project will work long-term or be successful without the support of local population, the local people and the community in those areas. But regarding funding, there's a little bit that I can share. So there's currently a Conservation Action Fund whose calls are now open, and they award 50,000 euros for conservation action grants, exactly to implement the kind of things that I was discussing. The Indianapolis Zoo just recently closed, the applications for the Indianapolis Zoo Saving a Species Challenge, and that was $1 million to promote conservation action that would show the best possible plan to change a species red list status from whatever red list it is to downlist it to the next best category or others. And that's a project that we're very happy to have supported, and it's something that I love doing as part of my work. There's also the Mohammed bin Zayed Species Conservation Fund, and I have the logo in the bottom, and they also do conservation action or welcome proposals for conservation action for inverts specifically. So all these projects would be open, all these funds would be open for conservation action on the ground to conserve fireflies or other inverts that might be in need of support. And this was all that I had to share with you today. Thank you so much for the opportunity to speak to you all and to, of course, learn from your experience. And I'm looking forward to support action-driven projects and to see fireflies driving in India and elsewhere in the world for many, many years to come. All the best, and I hope to see you soon.
Yeah, thank you, Professor Hendricks, for your very comprehensive presentation and the activities of the NGO, which you are running very actively in Indiana. Any questions to Professor Hendricks, please?
Not yet.
Any questions? Participants, if you don't have any questions to Professor Hendricks, we break for a small 5 minutes tea break. So we will have a break of 5 minutes, and then when we return, we'll be having a very interesting lecture from Professor Anurup Gohin Barua from Guwahati University, India. Professor Barua has specialized in firefly bioluminescence, and definitely he has extensive experience in this aspect of firefly behavior. So have a break for 5 minutes, and when we return, we will have a lecture from Professor Barua. Thank you. 5 minutes break. Respected participants, welcome back. Hope you could relax for about 5 minutes. And we will have the lecture from Professor Anurup Gohain Barua, Guwahati University, India. Professor Anurup Gohain Barua is currently the Professor of Physics, Guwahati University, and he has served the department head from 2013 to 16. Professor Barua's studies include propagation of laser beams through gaseous media, which involve stimulation of molecular spectra, spectroscopic characterizations on astrophysical objects like the comet and meteorite, on objects like bricks from cultural heritage sites, and on fruits. Professor present interests are firefly bioluminescence and thermoluminescence dosimetry. Professor Barua has published more than 50 research papers in peer-reviewed journals. He has guided PhD students on firefly bioluminescence, and Professor Barua is INSA's visiting fellow at RRCAT Indore. He is a Japan Society for Promotion of Science Invitation Fellow at Chiba University, Japan. Professor Anurup Gohain Barua, please.
Thank you, Dr. Chakraborty. Do you hear me?
Yes, yes, yeah.
And is my slide visible?
Yes,
Yes, sir.
yeah, yes, sir.
So thank you, and good afternoon, or good
Yes.
evening, or good morning to everybody.
Yeah, good afternoon. Yeah.
So first of all, I thank Dr. Chakraborty for giving me this opportunity to speak in this webinar. He has been doing a commendable work in making a platform of firefly workers for sharing knowledge on fireflies. I see that this is a webinar attended by mostly biologists. So in that case, I am the odd person out, because I am more interested in the emitted light and the pulses from the fireflies. And okay, so my talk will be mostly on the emission spectra and the pulses. And what I intend to know is that it is the light emitting system or the light emitting reaction in the life condition of a fire, because that oxyluciferin, that excited state, it is a volatile one. So only its analogs can be fabricated. So in the life condition of a firefly, the only information we can have is through the emitted light and pulses. So this is what I have been trying to know. And because it's a cold system, it does not emit heat. So had it emitted heat, maybe, I am not sure, maybe that could also have been used to understand some aspects of the firefly light emission. Okay, so today's topic is emission at the low temperature. So I start with a philosophical statement: the firefly gives light to its pursuer. We hope so, as we have been pursuing this. We have all been pursuing this. So these are among the most charismatic of all insects. So both poets and scientists have been inspired by this. So this is one of the poems. What should we make of fireflies? Their quick flare of promise and disappointment, their throwaway style. But of course, I am not here to speak on poems and songs. I am here to speak on something scientific. So as you all know, the bioluminescence means this is the process in which living organisms convert chemical energy to light. So too much of the reaction I do not wish to go. That the luciferin is the molecule, and the luciferase, which is an enzyme, it is the catalyst. So it is the oxidation of this luciferin molecule catalyzed by luciferase. So it is called chemiluminescence reaction. So just in very briefly, it's a multi-step process. So the trigger is oxygen. In contact with oxygen, that oxyluciferin is formed. And when it comes down to the lower state, the light is emitted. So this is very, very briefly, of course. So it is the most efficient example of a bioluminescent system. Its minimum efficiency has been estimated at 41%. So initially, it is thought to be about 90%, but the minimum efficiency value has been put somewhat recently, not that recent, it's 2008 SR photonics paper, that it is 41%. So most of the other light producing reactions are only about 1% efficient. So this is, of course, an efficiency which we simply can't ignore. This is an astonishing efficiency. So modern SR is clearly an efficient user of energy. So modern SR is the greatest scientist. Okay, so because today's talk is firefly emission at a low temperature, but prior to that, what we found at high temperature, it's a somewhat recent one. So you can say this is something like a precursor to what we are we have done now. So this is the Luciola, the Proy star, so which is widely available in the campus of Guwahati University, where I work. So this was so kindly identified by the well-known expert, Dr. Leslie Valentine. So this is the light organ or lantern, and for a male, there are two segments. This is Sclerosia substriata. It is found in a very small number in a bank of a very large pond in our campus itself, and again identified by Dr. Valentine. Okay, so what we found what we wish to found by this study is that at what time after sunset a particular species will come out, whether just after sunset or it will take some time after sunset to come out. So there is a classification of the fireflies depending on its time of coming out. It is early starting or late starting. The species which comes out in advance of 30 minutes after sunset . So these are called early starting, and the late starting ones are the ones which begin flashing after 30 minutes of sunset. So it is also mentioned that the dark active species, the peak emission, it is towards the green side. I mean, it is less than or equal to 558 nanometer. It is green, you can call. And once which begin its emission after half an hour from the sunset, which is the late starting one, it is a greenish yellow one, you can call. So this was published by Lal, Seliger, Bickley, and Lloyd in Science in 1980. Okay, I just want to see that why a certain species becomes early starting and a particular species becomes late starting. So you can see that the Luciola proista, its peak wavelength is 562. So obviously, as per the classification, it should come under early starting category or dusk active. And Sclerosia substriata, its wavelength peak is here at 558 nanometer, which is green. It should come under dark active or late starting category, as per the classification by those workers. Okay, so okay, we see at high temperature what happens. At 27 degrees centigrade, it is at 562 nanometer fine. At 42, okay, in between I have not shown here. It is just about at the same, just about to shift, and it has shifted at here. And at 45, you see that the peak comes here. And also, this comes at a full width at half maximum, meaning this is the maximum. And the half intensity is something here. So this is the width. This width and the half width at 42 degrees centigrade are approximately the same, the 55 nanometer. And at 45, you can easily see that it has become quite broad. It has become 70 nanometer. So it is called full width at half maximum, or simply half width. That half, it is a maximum. Take the half of that. So this is the width. So there is a shift towards the higher wavelength side at high temperature after about 42 degrees centigrade. This is what we found from the males of Luciola foresta. So it is mentioned that slight difference in the enzyme structure. So these are the reason for different fireflies emitting a slightly different spectrum, meaning that this luciferase, which is the catalyst, its structures have to be slightly different, and because of that, different species emitting slightly different spectral region. So because here at the high temperature, the peak has shifted towards the higher wavelength side. So the catalyst, the enzyme luciferase, must have undergone a structural change. So it possibly undergoes thermal denaturation, meaning that folding, the proteins are in the folding form. And chemists will of course be more comfortable here. So anyway, so I am also a physicist, not exactly a chemist, but then this is what I understand, that from the folding, it becomes unfolded. So it is at the 42 because the peak position has shifted towards the higher wavelength side. So there must be some kind of a structural sense. So we can say that it has undergone thermal denaturation. And about the flashes, the pulses that it emits. So at the normal temperature, say about 97, 98 milliseconds. So this is the time for which a pulse lasts. And when the temperature is increased, this duration becomes smaller and smaller. And at 42 degrees centigrade, it becomes a minimum. It becomes about around 50 milliseconds, you can call. And after that slight increase in temperature, it causes large increase in the duration. That is, at 42 degrees centigrade, the peak shifts towards the higher wavelength side, and also the pulses or the flashes becomes the narrowest. And this is the figure graph. From here, you can see at 42 it becomes minimum, and also the fluctuation you can see standard deviation. That is the deviation from the mean value. It becomes large, both at low as well as at high. So we can say that at 42 it is a special temperature. So the wavelength peak, it shifts towards red, and the pulse widths become the narrowest. So we can conclude that the enzyme luciferase probably gets thermal denatured at this temperature. Now about this sclerosia substrata. So here you can see at 28 degrees centigrade, it is here at 558 nm. But then at the 34, it just about begins to shift towards right. And then you can see that 35, 37, 39, 41, 43, and 45. The peak is shifted towards right. And the width is also become, or the half width, that is the full width at half maximum, it also becomes very wide. So these are the pulses here, starting from 25 degree. So where it is from about, say, the 180, very roughly. At 28, it is about, say, 170. At 32, it is about 130, 140. And at 34, it is about 115, 120 millisecond. So meaning that with the increase in temperature, the flashes become shorter and shorter. That is, it makes a statement about the speed of the reaction. If the speed is fast, it starts and ends quickly, meaning that the flashes have to be short. And if it is a slow reaction, it starts and ends slowly, meaning that the pulses are likely to be longer. So this is what we propose, because this is the only way to make a statement on the speed of the reaction. And this is after 34, which is the key temperature here. You can see it becomes now about 200 milliseconds. So earlier at 34, it was about 119, 120. Now you see just 1 degree, and it has jumped to 200, Then at 38, you can see 300 something, at 38, 400, and it was increasing. And from 43 and 45, it is no longer the flash type. It is just fluctuation of intensity, you can call. I mean, it's at the glow only. It is not on off type. So this is the figure. At 34 degrees centigrade, it becomes the minimum. Then for a small increase, it increases rapidly. So, and with the wide fluctuation. So 34 degree, we can say this is the maximum rate of luciferase luciferin reaction. And along with that, the peak also gets shifted towards the higher wavelength side, which we call as redshift. So we can say for sclerosia substrata, 34 degrees centigrade is high. For Luciola priesta, 42 degrees centigrade is high. And that is why Luciola priesta, it can come out just after sunset. There's no problem, but sclerosia substrata has to wait for some time. Temperature has to cool down. Only then it can come out. And that's why it becomes a late starting one. So, so this is the reason. So 34 degrees centigrade, it says that it has to wait for some time. Only then it can come out. Otherwise, it is likely to get thermal denatured, which is an irreversible process. So it also gives a reason for their choice of the habitat, because the temperature here at this part of the globe, it usually goes past about 34 degrees centigrade. I mean, in summer. I mean, even today, it has already reached 33, I think. 33 or 34, it has already reached. It has already become hot. So, sets from branches and leaves of the trees in the banks of the big ponds keep the temperature within the temperature optimum. So, maybe because of that, it requires a large pond, and as well as it resides in a tree, I think. So, in hot days, it is observed to start flashing 2-3 hours, sometimes more than 3 hours after sunset. So this is the reason. In fact, on the rainy days when the maximum temperature becomes 29 or 30, so it has seen to come out almost at the same time as Luciola foresta. So that is, there is little to choose in the appearance time. So this is what we found for the high temperature. Now our topic today is the low temperature. So what we can comment about a certain phenomenon. Okay, so we have noticed over a long period of time, over a dozen years of time, that the female fireflies have been observed to disappear a bit earlier than the males at the onset of the winter. Now, see, we have to think a lot about this, because ideally, both males and females should come together and live together. This is the law of the nature. But it's an observation of us that they seem to disappear just a bit earlier than the males. See, in this part, till the end of November, even in the first week of December, catching a few males is not much of a problem. But the females are extremely difficult to get even in the mid-December. Maybe 1 or 2 you can get. So, but most of them seem to disappear. And from about, but see, last year, at about 23 degrees centigrade, after searching for several days, I saw some light coming out from a hole covered with grasses and straws, some light. So I removed those and saw that it's a female. So it is not exactly a hibernation you can call, but almost like a hibernation. Maybe a flying male will see the light and it crawl inside, and a mating might occur there. So seed doesn't come out. So in that case, we can say it's a kind of semi-hibernation time. Okay, we will see why the females disappear a bit earlier generally. Okay, Dr. Valentine once informed me that based on females, identification cannot be done. But as this place is inhabited by Luciola foresta fireflies only, we can say that the collected females belong to that species. See, I belong to the physics. I don't know about those taxonomy business. Only Dr. Valentine can tell them. So this is the male. This is the lantern. This is the female, and you can see there is only one light emitting segment here. Okay, so it's an earlier finding from our group that you can see at 30 degrees centigrade, a male pulse width, this duration is about 97 millisecond. And for a female, it is about 190, 195, something like that, meaning almost twice as long. Male flashes are almost twice as long. Sorry, the female flashes are almost twice as long as the male one. So what does this imply? That this implies that the light emitting reaction, this chemiluminescence reaction, it is a slower proceeding reaction or a longer lasting reaction. So see, I repeat that Mother Nature is the greatest scientist. So that the female flashes are longer than the male. She must have a reason. Mother Nature does not do anything arbitrarily. So I leave it for you to speculate on this. See, here you can see at low temperature, this blue shift we can see that is towards the lower wavelength side. At 26 degrees centigrade, you can see it is here. And 20, 15, etc. coming at 11 degrees centigrade, you can see it makes the shift towards blue. And at 8 degrees centigrade here, this is for male. And for female, it is observed at 15 degrees centigrade itself. This is the 15 degrees centigrade, and this is 11 degrees centigrade. See, it is shifting towards the lower wavelength side. And for another one, another male, you can see here at 11 degrees centigrade, it shifts here, the 8 degrees centigrade. For female, it is at 15 degrees centigrade, that is at a significantly higher temperature, it gets shifted towards the lower wavelength side. And this is just for 12.5 degrees centigrade. Okay, so it normally appears at 562 nm, almost the same for both male and female. But then at 11.5 to 11 degrees centigrade for male and 16.5 to 15, that is about 5 degrees centigrade or the 4 degrees centigrade more, the female peak gets shifted. So we can correlate with their the low temperature tolerance. That the low temperature tolerance of males have to be better than that of the females. So a blue shift of approximately 5 manifests for all specimens used in the experiment. So about the 25 hits of a male and female we used. So this is unlike in the high temperature one, this is a reversible. So when the temperature is increased, it becomes 562 nanometer again. So just a repetition. So the slight difference in the enzyme structure is a reason for different species emitting slightly different spectral lesions. So because the peak is now a defined one from the normal temperature peak, so structures of male and female firefly luciferases must have changed slightly. So we could infer that cold denaturation of enzyme luciferase probably occurs for males at 11.5 to 11 degrees centigrade and for females at 16.5 to 15 degrees centigrade. So it is the light organ, a simple photograph at 11 degrees centigrade for male and at 15 degrees centigrade, you can see the greenish tinges come here, whereas for male, it required a considerably lower temperature for the greenish tints to appear. And this is at 9 degrees, you can see it has become the predominantly greenish, and it is for female. Otherwise, it is predominantly yellow at the normal temperature. This is some visual indication. So these are the flashes. I just wish to go fast here. So here, see with increase in temperature, a decrease in temperature, the flash duration increases, then it gets split at low temperature. So obviously, its duration will increase and randomly. So statistical analysis is does not appear to make much of a sense, but we did some kind of a statistical analysis also. So the duration will increase considerably, and for the male from about 11 degrees centigrade, and it increases considerably. So you can see that we started with 145 millisecond at 26, it becomes 1696. Then see, you can see it goes up to, it's considerably large. So these are again the same for different male specimens showing large deviation from the mean values. So this is how it changes. And the abnormals increase with considerable fluctuation, that is standard deviation also, it becomes quite large from below 20 degrees, 12 degrees centigrade. And this is for female. You can see at the normal temperature about 200 something millisecond. And when the temperature is lowered, it becomes more and more, it gets split here, and it increases considerably, so it is like that. So up to the 14 degrees centigrade, it is shown here. Because the females stop flashing less than about 14 degrees centigrade. It is very difficult to get a response, meaning she cannot tolerate such low temperature, unlike the male. So it is for different specimens at the same temperature range. So sorry, this A should be, it is 26 degrees centigrade, not 16 degrees centigrade. I'm sorry for this. It should be 26, 20, 17.5, etc. is coming. So starting from 220, it becomes a 1067. So at the peak 16 temperature of about 16 degrees centigrade, there is considerable increase. So this is how we can plot for male. You can see that this standard deviation, considerable increase in the duration, plus fluctuation is also considerable from about 15 degrees below, but then for male it is from about 11 degree, you can call around this. The fluctuation is large, and this is for female. So again, from about 16 degrees itself, the fluctuation is more, and considerable increase in the duration. And this is the table, but then without looking at it in detail, you can see at 12 degrees centigrade, the flash duration, it becomes considerable, and fluctuation, especially 11.5. You can see flux standard deviation, that is the deviation from the mean value, it is so large. So something must have happened at 11.5 degrees centigrade. So this is for male and this is for female. You can see from about 16.5 degrees centigrade, the fluctuation is large and all, but then it is not as striking as in the case of male, but is still considerable. And so at 15 degrees centigrade, you can see it is 1030, and with fluctuation there, and also number of flashes is also decreased, meaning a few female fireflies emit at such low temperature. So abnormal increase in the pulse width with wide fluctuation along with the phenomenon of blue shift apparently supports the proportion of cold denaturation. So till now, only 4 proteins are reported to undergo cold denaturation above 0 degrees centigrade. So these are, well, the YFH1, then C-terminal domain of L9, and scaffold protein for FES cluster in Escherichia coli, then HIV1 protein. So we propose addition of enzyme luciferase of this firefly in the live condition protein. So only 4 till now. It could be 4, but then to be absolutely conclusive, some thermodynamical studies have to be carried out on the firefly luciferase. Only then it could be conclusive, but that one could be left to the biochemists. So here in mid-November, the late night temperature goes down close to the temperature of possible cold denaturation of female. So that is why one has to be very lucky to get a female at that time. So for males, it is only about 11 or 11.5. So appearance for them is not so difficult. But yes, their number also decreases at this time, but not as much as the female. So the rare, a male could be seen flashing even in the first couple of days of December, just after the sunset. So as males normally flash in search of females, a female or two might be there. But in our observation of a dozen years, no female has been sighted. So I think this is a finding or observation from the unique to our group. So we myself have doubts because necessarily that males and females should come together and live together. Why the female should disappear earlier? So we got the answer last year when, as I have already narrated, that I saw some light coming out from a hole about 1 foot or 1.5 foot deep, covered by grasses and straw, and that was a female. I mean, she could not come out to the surface, just stayed inside. So if a male sees that, he could go crawling inside, and okay, this mating can be done there. So just to conclude, so at the peak shifting temperature, of course, this transition probability, this is some physics. Okay, I want to skip this one. So the lower value of this peak shifting temperature for males imply their greater tolerance of low temperatures, and this could be the probability for them generally disappearing later than females when the winter sets in. Okay, so thanks to Department of Biotechnology and Science and Engineering Research Board for granting projects. Okay, and thank you very much for a patient hearing. Thank you. So patiently listening to this. Thanks from my side, and special thanks to Dr. Chakraborty.
Okay. Thank you, Professor Barua, for your in-depth study, and you have covered many aspects of bioluminescence, and it's one of the first of its kind study, in-depth study on bioluminescence. I'm sure that the participants will have some queries or questions. Any questions or queries to Professor Barua, please?
Okay, I think that this kind of study should be carried out on all the species of fireflies.
Yeah.
So some kind of a collaborative approach is definitely needed, especially in India.
Yeah.
So because only then the final picture will be clear.
Yes.
Because you see that this excited that molecule, it is volatile. So about this light emitting system or the light emitting reaction in the life condition of a firefly, if we have to get any knowledge, it is through the emitted light only. Otherwise, you have to kill the insect. So that way, that the normal condition will not be there.
Sure. Angle. Okay.
I have seen one question. What are the major instruments used for this study? Okay, you require a high resolution spectrometer. So we use that HR 4000, that spectrometer from Ocean Optics, USA. And for the pulses, you require an oscilloscope, digital storage oscilloscope, and the photomultiplier tube is needed. The photomultiplier, because you see that the firefly light is a very big one. It has to be amplified. So a photomultiplier tube, and that is a waveform, the pulses or flashes, it can be recorded in a digital storage oscilloscope or DSO. So these are and also to monitor this temperature. Okay, you can use a thermometer also, but it will be better if at the location of the firefly, you keep a small IC, a temperature sensor. It is LM35 is the one. LM35 is a small IC. So you can keep it close to the firefly light organ. So almost the same temperature you will get. It's a small one. Or a thermometer also you can use, but this IC LM35, a temperature sensor, it is the ideal one. I mean, as far as I am concerned, what I think.
Professor Leslie Ballantyne, Madam, you would like to have any questions? Professor Leslie Ballantyne, Madam, have you any questions?
Hang on, I've just unmuted. I just unmuted myself. I've been trying to think. Dr. Barua said that he didn't understand the taxonomy part of it, and I am having a lot of trouble with the physics and the chemistry part of it. But I wonder whether there are aspects of what we know about the biology of these species that we might be able to put together some sorts of other explanations, or things that we can see that we can now say, okay, yes, I can see why that happens now, because Dr. Barua has shown me about what happens with the luciferase being denatured and things like that. But that's about the limit of my expertise.
Yeah. So that's why a collaboration is needed, because the taxonomy part you have to do, because we don't know anything about this. For us, it is simply a biomaterial. So you have to do the taxonomy part for us, and then about the optics part we can do that.
Yes, because there are two different aspects, and definitely will be yes, and Professor Barwa, you are most welcome to the South. You are most welcome to the South, and I hope you are planning to visit us, and we can take you to different kinds of landscapes where you can come across different species of fireflies, and you can study them from the bioluminescence aspect.
So thank you very much, Dr.
And as you said, that we are also very good in taxonomy, and definitely we need the guidance. We will be very fortunate to have our guidance in this sort of study. So you are most welcome. Please do plan and visit us. You have already written us a letter, and we are happy that you would be visiting us soon. There is one more question from Dr. Ved Prakash Singh. What type of light should be recommended? What type of light should be recommended for vehicles traveling at night? What type of light should be recommended? And we have also seen that Dr. Chandana from Sri Lanka also had given some photographs as to how the beam rays should focus and what should be their shape. So can you just enlighten on this aspect, Professor Barwa, to the participants?
Yeah, about this light of the vehicle, I cannot say with a certainty, but then, okay, if we are concerned with this firefly, so I mean, better not to have any light at all, because in our campus also we have seen earlier this sclerosia substrata. So those used to reside in 2-3 trees near the bank of a big pond. In fact, 2 big ponds are there, very large ponds. Okay. But then after that light, the street light is given there, erected there. I have seen that those have gone from there, and they are now found in some other place now. A nearby one, of course, but they have lived. So I think better not to use any vehicle in the place where these fireflies are found. It is better. It is better. I mean, as little as possible. Otherwise, these fireflies, they seem to be repelled by this light. You know, they cannot tolerate or withstand this light. This is what I have observed over the years.
Yeah. Correct. Okay. If any more questions to Professor Barua, please, from participants? Any more questions? Then we will have a lunch break now. We break for lunch. Thank you so much, Professor Barua, again, for your very in-depth study on bioluminescence. And definitely we'll be able to collaborate with you anytime we are ready, and you can plan accordingly.
Thank you.
Dear participants, we break for lunch now, and we will be again at 13:45. That is a lunch break for about 45 minutes, nearly 45 to 50 minutes. There's one more question from Ved Prakash Singh. Professor Barua, can you answer? There's one more question. Can we take one more question, Professor Barua? Hello.
What's the question?
Yeah, the question is, can we enhance the capability of Fireflies to resist vehicle lights? Can we enhance the capability of Fireflies to resist vehicle lights? That is
Oh,
the
that.
question.
That only the biologist can tell, but I simply analyze that emitted light. So about that, I am not the expert, so I cannot answer that. So,
Okay. Thank you then, all of you. Thank you so much. Please don't miss the other important lectures we have post lunch. So we break for lunch here and we'll be back at 13:45 sharp, please. Have a nice time.
Okay.
Good afternoon, all the participants. So it's 1:45 Indian Standard Time in India, but in other countries, the times will be different. So a very warm welcome to all of you for the afternoon session of this important webinar. Ladies and gentlemen, we have with us Dr. Ramon Guzman from Spain. Dr. Ramon is a naturalist who became fascinated by glowworms during his childhood in Granada, Spain. He has studied forest engineering and holds a PhD in agronomy. In 2008, worried about the status of these insects, he launched a citizen science Firefly survey web project in collaboration with Rafael de Kock. Have you ever seen a glowworm? Has increased the knowledge about lamparets all over Spain. He is going to tell us about fireflies in Spain. Professor Ramon Guzman Alvarez, please.
Sorry. Thank you very much, Dr. Chakrabarti, and all the people who are following this very interesting meeting about fireflies, glowworms, and all these kind of insects that are so fascinating for all of us. Well, as Doctor has said, I'm working in fireflies during the past 14 years here in Spain, and with the goal of increasing the knowledge that we have about these insects that are very well known by every people, because they everyone knows this kind of insect because of this particular behavior of the bioluminescence. But when I started with the research, there was very little academic and knowledge about this group of insects. So with Rafael de Kock, that he's the very best the very best fireflyer here in Europe, we launched a website citizen project called Have You Seen the Glowworm? And during these years, we have been working, collecting records and collecting other information in order, as I said, to increase our knowledge. We have now results of all these years of investigations, and as I am going to expose in this talk, we think that we can go in other phase of our collective research about fireflies. So I'm going to share the screen. I hope not to have problems about this matter. Wait a minute. So let me share the presentation with you. Sorry. So let's go on, and we can start with the presentation. I think that you can see the presentation.
Yeah, we can see.
Yes, okay, thank you, Doctor. So let's start with its title is Knowledge and Monitoring of Glowworms and Other Fireflies in Spain. First of all, I would like to thank to the organizations of the meeting for inviting us to expose our results, and of course to the International Firefly Network that has been doing the huge work with the goal of sharing the things that researchers, but also other people, are collecting about the behavior and other information about this insect. So let me first of all, please make you a brief introduction, because this meeting is a global meeting, so every one of you belongs to different countries, and I would like to show where we are living and some characteristics that are important because influence our fauna of fireflies. We are talking about the Mediterranean Sea region in Europe, in the western part of the Eurasiatic continent, and I guess that most of you have some reference about this area, because it's very well known in all the world about our landscapes. But we're going to move specifically to the western confine, to the Iberian Peninsula, shared by Portugal and by Spain, where we have standard Mediterranean landscapes. I mean that composed by quercus, olive trees, almond trees, and coastline, mountains. It's very mountain regions, but we have climates going from the desertic or almost desertic zones to high mountains, climate areas, and even super humid climates. And we are a very, very long history of sharing the nature of man-made use of nature with livestock of different species and race, goats, pig, cattle, and so on. We have different kinds of forest landscapes, from fir to pines and to quercus and to other species. And of course, we have an extensive agricultural areas composed of different products. Of course, we don't have some things because our latitude geographic situation. Of course, we have not a vast surfaces of natural territories because we are living in a man-made territory for a very long time. And saying all of this, that maybe you are asking, well, but what about us? What about the fireflies in this context, in this geographical context? Well, we are not used at a wondrous spectacles of thousands of fireflies brighting together at the same time, as is more or less usual in other countries, in North America, or in Asia, in the Pacific Island, and so on. It's not our fireflies history for saying. We are more used to this. I mean, our fireflies has very, very, very modest insects that usually light alone during the first hours of the night, or maybe in the leader cluster of females lighting with the purpose of searching for a male. But in our society, our child and our adults know very well and love glowworms, although it's more usually by this time that people don't really contact with living fireflies, but more with cartoons or with TV movies reference, or with puppies like this, because in fact, it's not such a matter of being disappeared, this species, as I'm going to say, but it's most of all a matter of that people are being far and far away from nature in their usual living. No? So we can't hardly see unless identify the handful of species living among us. We have, as I said, a very, very discrete number of fireflies species, and with the natural history that that make them a little bit, if not difficult, at least a bit hazardous of finding in nature. So, in fact, the kind of glowworm that is most probably to see by us here in Spain and in other countries of the Mediterranean region is something like that. It's a bug, it's a beetle. This is the male of Nyctophila roici, one of our commonest species. And this is more or less usual to find out just during these weeks, approaching to lures, to artificial light, because they are at certain point confused by our artificial lights. And also, it's usual that we can find this kind of fireflies during our walks in nature. This is the larva of Lamprilis, but people can't associate this bird with the adult, with the female that emit the light, because the larva of Lamprilis is just another bug, like a worm that is creeping on the soil. So people usually don't know this, but these birds are certainly glowworms, not female, not adult stage, but it's more frequent to find this stage because the female time to be seen is only 1 or 2 weeks during June and July in the year, and the rest of the time is more usual to find the larva or even to find the male during June and July. So as I said, some years ago, in 2010, Rafael and myself launched a way to unveil the hidden life of a Spanish glowworm. The project is called "Have You Seen the Glowworm?" Has visto una luciérnaga in Spanish, and it's a website where citizens, where people can send us photos, records, and other information about the glowworms they have seen, and we identify them for people, and by this way we increase our common knowledge about this. So we have gathered thousands of data and achieved some conclusions during this year, as I said. And just during the past week, we have launched the review of all our results in the way of a scientific publication called Catalog of the Species of the Family Lampyridae in Spain. And it's something like a baseline of knowledge about this group of insects that in fact we have the need of it. During our experience like fireflies, we have to answer many questions for many people interested in the glowworms. Here you have our audience, our public in one of our seminars. There are plenty were plenty of childs, of boys, very interested in what I explained, because as you know, fireflies are very loved insect for everyone. But one of the most asked question is the million-dollar question, in my point of view, about fireflies is, what can you do to have glowworms in my garden, in my orchard, or in my field? This is one question that people are very interested in, because people have the remembers when they were child and they thought that fireflies were more common and they could see them with their grandfathers and grandmothers at the countryside when they were they were bored or something like that. So people, now that they are adults, they wanted to return to these remembrance. And one of the way that they think that it could be possible is to bring glowworms to the gardens or fields. But do you know what we answered to this question? Well, we said, well, first of all, you have to try to work slowly, just looking calmly as you did when you were a child. So you will have the chance to observe the beauty of fireflies. We are living in a different society than 40 or 50 years ago. We are very, very more busy, and we don't do the kind of things that we used to do when we were a child. I mean, for instance, to walk alone for a while during the night time, and this is the kind of situation that is suitable to find glowworms in nature, and preferably, of course, walking at twilight or at early night time for increasing the chance to find it. The second thing that we answer is that it's important to take care of our garden or field, and of course, and the snails that live there. So improving and let the habitat be pleasant for nature. I mean, for glowworm nature. It's obvious if you have a garden and you employ a lot of insecticides to control snails, it's not good at all for fireflies, because glowworms, when they are at the larva stage, they eat snails. So we have to be careful with our near nature in order to build the suitable conditions to maintain our populations of lampreys. Because you are changing all your nature with glowworms, and maybe you don't know. So you have, so the thing is that maybe you have the insects there in your garden, but the point is that just you have not the chance to see it, to look at it, because you don't walk at the right time, or maybe you are not doing the thing at a proper way to improve the habitat condition for them. So we can't, and I said, I answer to people asking this question, so I ask that we can't bring to your garden glowworms, but we can help you to try to find them. And this is the point. For us in Spain, it's important because, as I told you, we have not cluster of thousands of glowworms. That's very, of course, easy to detect, and people, moreover, researchers know very well where this cluster. No, the chance of finding glowworms in Spain is hazardous. So what we have to do first is to look at it at the proper moment in a proper way, and the second thing is to maintain the habitat conditions suitable for these insects. So the key point is to look for them at the right time and at the proper way. And the other, because one thing that we explain continuously to people is that we need to know why glowworms glow. And this is, of course, you know, the reason is because they are more or less, this is the way to be love one to another, and to make the courtship, to find a partner to their reproduction. And this is in Spain mostly a matter of females. Although we have also as well some species where males breed in the courtship. I'm going to explain this issue after. most of our species is the females that emit the light in order to look for the females. So this is more or less how this works. This is the female, in this case, the species Nyctophila roici, and this is the male. The male, as I said, is a beetle, a winged beetle that can fly, and the female by twilight or at the early hours at night, bright, and the males approach them, and finally the courtship is made. But sometimes it's not all that simple, because although we are used to the females that emit the light, in our species also the eggs can exhibit bioluminescence, even the larva and even the pupa. But of course, these states are not very frequently seen as the adult states. Well, let's move to the catalog, to the relation of the fireflies that we can find in Spain. This, as I said, the most, I guess, the most frequent stage that can be seen, because it's the larva stage. During 2 years, larvae move on the ground looking for snails and eating them. But as I said, the people don't connect this bug with a glowworm or a firefly. It's more usual to detect the males approaching to our lights, and what we like to see, because of the fascination of the light, is of course the female. This female is the most common in Spain, Nyctophila roici. This is the second most common, is Lamparis iberica. Well, following the work of Owens, Van den Broek, de Kock, and Lewis, is a very interesting work called Behavioral Responses of Bioluminescent Fireflies to Artificial Light at Night. They systemized the fireflies in Europe in 4 groups according to the courtship behavior. And it's very interesting to show these groups in order to understand how our species are and interact in the nature. So the first group, the A group, is called the dark species, are species with diurnal adults that do not employ bioluminescent courtship signals. Instead, females are known or presumed to have pheromones to attract flying males, which have elongated antenna to look for these pheromones. The group B is called the glowworms, the royal glowworms, and are sexually dimorphic species with brachypterous or apterous females, so females that can't fly, which start courtship emitting continuous glows while perched near the ground. Males are flying around and searching for females' glow signals from the air. This is the group B. The group C is called roving flashing. This is the fireflies, really, and both sexes bright using discrete flashes of light. Females of this species are often sedentary, so maybe they are capable of flying. Flying males advertise by repeating a species-specific flash pattern at regular intervals, and females answer with response flashes in sound that we can define as a dialogue between males and females. And finally, the group D is the synchronous fireflies, that males of this species form conspicuous legs, congregating thousands of them each evening, which in particular display trees. Males collectively signals by flashing together in synchrony, and the flying females go toward display trees from surrounding these trees, and these fireflies have been very common, have become very popular because they are becoming a tourist attraction. Well, knowing these 4 groups of firefly glowworms and fireflies, let's know what are the species that we can find in Spain. Well, first, the group A, so the dark species, we have two of them, is Phosphaenus hemipterus and Phosphaenopterus metzneri. Phosphaenus hemipterus is a species more or less presented all over Europe, in the north part of Europe, and we have found very, very few records here in Spain. It's a species that doesn't emit light, so it's difficult to detect, and so we think that this is one of the reasons that is very, very little knowledge about the presence of Phosphaenus hemipterus in Spain. The second species is Phosphaenopterus metzneri. It's only found in a very little geographical zone in Spain and Portugal, and some authors think that it's only a variety of Phosphaenus hemipterus with more elongated wings. Moving to the B groups, let's show the glowworms species. We have the Lamperis iberica. Lamperis iberica is a species presented to science some years ago, and this is more or less, it is very, very close to Lamperis noctiluca that we're going to see after, and this the very usually present in the north part of Spain. This is Lamperis noctiluca. Lamperis noctiluca is a very widest distributed species all over the Eurasiatic continent, and we have population in the north of our country. Here is Nyctophila roechi, the most common species here, and this is related to Mediterranean areas, and you can find it from almost desertic zones in the coastal line, and but also at the high peaks of our mountains. This is Nyctophila haydeni. This is an endemic species of the Balearic Islands, related to Nyctophila roechi, but also to Lamperis species. And we have also 2 very little species of Lamproiza paulinoidea, Lamproiza mulsanti, that are species with light behavior more or less similar to Lamperis, but they light at an early moment in the year, during the spring month. Instead, Lamperis and Nyctophila are lighting during June to August, and these species have the courtship during March from to May. They are very little, so that's one of the reasons that are not very known. But although they light during night, of course, the females, they are not very, very frequently seen by observers. This is Lamproiza mulsanti, it's related with the former. And here we have one of species of the C group, so the robin flashing, the fireflies, that is called Lysula lusitanica, that at the moment we have only records from Portugal, but not from Spain. we suppose that it was a species that was extended through Spain in the past, but by the moment we have not been able to detect Lucera lusitanica again in our country, but we suspect that we will do in the future, of course, thanks to our collaborators. This is a species in which the male bright fly while flying, and the female wait for males emitting light, perching on leaves or something like that. And finally, we have an exotic species that have arrived to our country from South America. We have discovered that in the origin geographical area is Argentina and Uruguay on the south part of America. It's called Photinus gnaticolis, and at the first time we detected it with other researchers at the northeast of Spain, on Catalonia, the province of Girona, but also we have found a small population isolated in Extremadura on the south of the Iberian Peninsula. This is, as I said, an exotic species that is spreading to the north, and is just during the past 3 years have passed the Pyrenees. I mean the frontier, the mountain frontier between Spain and France, and now they have a population in the south of France. We are monitoring the expansion of the species, and although it's very, very, very spectacular because the flash behavior during night is very attractive, we are a bit worried about the duration and the impact to the environment that this species may have. So we are monitoring or trying to monitoring, let's say, these species. And lastly, of course, only to say that we don't have species belonging to the D group, to the species that are clustered in synchronous, and that's all. So now, and I'm finishing the presentation. We think that it's time to increase our knowledge through other approaches. So we've got, as I said, the baseline of knowledge about what species we have in Spain, what are the distribution, and things like that. We have the support of citizen science web project that collects records from all over Spain. But we need further information to determine, for example, the factors, management techniques, landscape context, and so on, that determine better conditions for growth walls, or, for instance, the temporal evolution of selected populations, or something like that. We have to move on to a specific fieldwork-based research. And how can we do it? So some ideas could be gathering people interested in being firefly researchers at different levels, of course, using standardized methods to sample populations under scientific supervision, of course, and designing procedures to obtain relevant and comparable information, or obviously at proper date, in Spain mostly from the early June to the end of July. So we have a lot to do. So let's join to this global knowledge experience. And thanks very much for your attentions. And we'll see looking for glowworms all over the world. Thank you very much.
Thank you very much, Professor Rahman. We had a beautiful glimpse of a beautiful country, Spain, and the hidden life of fireflies. Any questions to Professor Rahman from participants? Participant, please, if you have any questions to the professor, you may please proceed.
Yeah, Dr. Chakravarti.
Any story? Yes, yes.
Yeah,
Yes,
I want to know that how much response from the community for the conservation of fireflies in Spain?
Sorry, how much?
How much contribution the community is doing?
You mean or the society in general terms?
Yeah, under your citizen science program.
Yes. Oh, yes. It's a key point because not all our knowledge, but most of them is gathered through the records that, for instance, people upload to City Science web platform as e-naturalist or things like that. But it's not a matter of people being very interested in fireflies, but of people being interested in general in natural history of insects or in collecting information about nature. So in our country, it's not people love fireflies, people love glowworms, people are interested, but normal people, but they are not research or investigation groups about this. So it's not an issue belonging to normal, I think, or standardized research. Maybe I answered your question.
One more question, Professor Rahman. So far, there are any plans to regulate?
You very much. A very wonderful presentation and knowledge.
Yeah, thank you.
Yeah. So far, are there any plans to regulate Photinus signaticolis populations? I think it's already spreading through France.
Yes.
What's your opinion?
Yes, we are working together with French researchers about this issue. In France, they are very worried because it's because the climate is more suitable for these species. At least we suppose that in Spain, because we have more drier condition. So in France, they are very worried, and they are working with authorities in order, first, to monitoring, as I said, the population, and second, to try to prevent the man-made spreading of the species. Right. You think that there are two kinds of possibilities of spreading. One of them is the natural movement of the species, because it's a species that also males but also females fly. But there is also the possibility that people wanted to have this kind of species in their garden. So this is very, very, very crucial point, and very worried, because it's a way to contribute to spread the population of the exotic species in a very worrisome way. So we are, the answer is that, in fact, we are worried, and we are working with French people in order to try to know much better and to prevent the expansion.
Okay, sir. Thank you so much, sir, for your very informative presentation and have very knowledgeable documentation, very extensive documentation of fireflies in Spain. So thank you so much, sir. From Spain, we move on to US now, and we have with us Dr. Christopher Crossley, who is a professor of biology at Fitchburg State University and chair Fireflies International Steering Committee. He earned his bachelor's from Brown University and PhD at Tufts University. His doctoral research focused on the selective and adaptive role of female mating preferences for flash signal characteristics in fireflies. He continues to study the evolution of firefly flash signals, including through the interaction of fireflies and potential predators. Dr. Crossley also partners with the Massachusetts Audubon Society on Firefly Watch, a community science project monitoring firefly populations in North America. So we are very thankful to you and very grateful to have you with us today. Please, sir.
Thank you very much for that wonderful introduction. And thank you also for putting together such a fantastic program. It's really been very, very informative and really inspirational for me to learn so much about fireflies, certainly from great speakers from around the world, like we just heard from Spain, but also so much about Indian fireflies in particular, an area where I have to admit I've been woefully underprepared. So as you noted, I will mostly be focusing on North American fireflies and focusing in particular, as I share my screen here, focusing in particular on, there we go, firefly flash behavior. I hope in the process of this talk to convince you that there's a lot left to learn about firefly flash behavior, and that as you study fire, and as all of us continue to study fireflies around the world, there's some really interesting insights we can continue to gain, not just for the purposes of science, but also as a way to further our conservation efforts that so many have been talking so eloquently about over the past couple of days. I'm going to take a brief moment before I jump right into this and do a couple of sort of self-serving things. The first is to just encourage you to go ahead and consider, if you haven't already, joining Fireflies International. This is a really wonderful organization that does a variety of things, but including putting together every 3 years or so, a regular international firefly conference, our next one coming up in 2025 in Mexico. So I really do encourage people. I've put the website and the URL up, and I encourage you to check that out. And if you haven't already, I know many of you probably already have engaged with it, but that's my first little plug there at the beginning. And the other plug is a little unusual for me. I usually keep business and personal things fairly separate, but given that this is there's so many people attending this from India, I wanted to, and I will be traveling to India this winter. I wanted to just highlight, this is my family here. My wife is from Kerala, and these are our 6 kids, a wide range of ages from 6 months old to 25 years old, but we'll all be in India. And I am so inspired by learning about the fireflies of India. I hope that even if it's just by email, I can start to connect with some of you wonderful researchers in India to learn more about what you're doing, so that as my family and I, travel to India, I can stop having it just be personal stuff, but maybe even include some research in it as well. So that's my quick plug on a personal level, and let's jump into the talk. So I'm going to, as I mentioned, have 2 major themes here. The first one is talking about the evolution of flash behavior and really highlighting sort of 6 personal experiments that illustrate, I think, some of the range of things that we can learn about flash behavior and the evolution of flash behavior in fireflies in a variety of different ways. And those fall into two big groups. One is the selection on flash signals as signals to conspecific, signals to other fireflies of the same species. And the other is as selection as signals to potential predators. And when we talk about the signals to potential predators, there are, of course, cases, especially in the Americas, where these signals are beacons for predation by other firefly species, but also opportunities for these signals to function as aposematic displays to bats or other potential predators. So I'll talk a little bit about each of those things. And then I want to conclude coming back to this theme of conservation. How does knowing more about firefly flash behavior help us to inform our conservation efforts? I'll talk about the pitfalls and promise of 3 different efforts here in North America at monitoring fireflies and getting community science programs going that engage the community in. And then I'll just conclude with a few thoughts about outreach and education. So let me jump in to this with a quick and admittedly biased and incomplete phylogenetic tree here from a paper my advisor Sarah Lewis and I wrote many years ago at this point, I guess. And at the time, this, as I said, incomplete phylogeny was meant just to illustrate a simple idea, the idea that there is this diversity to the ways in which fireflies communicate with flashes. And on a simple level, that can be the difference between fireflies that actually produce discrete flashes versus fireflies that just produce long glows, or even fireflies that don't use flash signaling at all. And I present this to begin the argument that firefly signaling behavior probably is very important in the evolution of the diversity of fireflies we see out there. Because there is this diversity of ways in which fireflies communicate with bioluminescence. If we zoom in to one particular group from North America, the Photinus fireflies, our most species-rich North American group of fireflies, we can also see that even for flash signaling species, there's a great deal of diversity to the ways in which they produce those flash signals. And this is an incomplete list because there are many other Photinus species in North America, but it's a list that was abridged in order simply to illustrate the idea that some Photinus fireflies produce single pulses. These are the male flash patterns in this column. So single pulses repeated at regular intervals, and those pulses can vary in their, how frequently they're produced and how long the duration of the pulses are. And then females respond to those, usually with a single pulse of their own after a species-specific female response delay, and that's illustrated in the right-hand column. Now, I've highlighted 2 of the species that I'll be talking about, Photinus ignitus and Photinus greeni, and they illustrate one of the important differences that occur besides just the rate at which flashes are produced and the duration of those flashes, but also the fact that for Photinus species, we may see species that produce pairs of flashes. Photinus greeni is a species that produces their flashes in pairs and then repeats those pairs of flashes. And Photinus ignitus is one of the many species that produce just the single pulses repeated at a regular interval. There are, of course, even species like Photinus consimilus and Photinus carolinus that produce trains of multiple pulses, not just pairs of pulses. And for each of them, again, females produce usually a single pulse that is produced at a species-specific time delay from the male's first pulse of their flash pattern. Now, to further orient us to this diversity in flash pattern, I just want to introduce a few terms. So we've already talked about the idea that there could be single pulse, double pulse, or even not shown here, multiple pulse species. And the idea then that those pulses could have a specific duration, that if pulses are produced in pairs, there might be an interpulse interval. And then certainly, whether they're produced singly or in pairs, there's a flash pattern interval, the rate at which you see either a single flash or pairs of flashes repeated. And then finally, the female response delay, as I mentioned. And one of the places I began my research was examining, because in Phytinus fireflies in particular, but in other North American fireflies as well, we have this characteristic female response delay. We can really use that as a tool to examine, are females putting selective pressure on the evolution of male flash signals by preferentially responding to male flash signals that have certain characteristics? And can that, as a result, perhaps help us to understand the evolution of the diversity of flash signals that we see in a genus such as Phytinus? And here's an example of that for both Phytinus ignitus and Phytinus greenii, what I call experiment 1 of the 6 experiments that I'm talking about in this particular talk, where we could present females of Phytinus ignitus in this first figure here, figure A, with single pulse flashes produced at a range of different pulse durations that correspond to the range observed in the wild in this particular species, or actually not in the wild, observed in captivity in this particular species. And so this bar here represents the range of flash durations observed. The triangle here represents the mean. And we can see that across that range, females tend to show higher preference for some of the longer duration flashes and respond at a lower rate to the shorter duration flashes. However, this pattern is reversed when we look at a double pulse species like Phytinus greenii, where females are in fact showing lower and lower responsiveness as pulses increase in duration. And in fact, pulse durations weren't even tested up at the upper range of the naturally occurring pulse durations seen in the lab. And what they really seem to be responding to, a really steep response to females showing increased responsiveness when there is a short interpulse interval. So remember, again, that's the time between those two pulses, because this is a species that produce paired pulses. And so females seem to prefer that a really fast pair of pulses. And we can imagine that this kind of difference in female preference, if it evolves between species, can really drive differences, the evolution of differences in male flash patterns. So that's one interesting thing for us to look at as we try to study and understand the diversity of flash signaling in fireflies. We can also examine, and that's what we'll talk about with experiment 2, whether males are more or less attracted to different female flashes and female response flashes. And so in this case, I used an instrument in the field that we developed with the support from a National Science Foundation grant that we called virtual firefly instruments. We could program them with a computer and then set them out in the field, producing a characteristic flash. And in this case, in a field full of Phytinus ignitus males flying around, we could produce Phytinus ignitus female-like flashes at a range of different durations, either the standard mean female duration that we'd measured, or the mean plus 100 milliseconds, plus only 50 milliseconds, minus only 50 milliseconds, or minus 100 milliseconds, and see, were males more attracted to, by observing these circles in a number of the upcoming images you're going to see and experiments you're going to see represent little platforms that we created out in the field. And what we observed was the numbers of males approaching and trying to court these simulated females that we had out in the field. So we would have student observers around these different platforms counting the number of males that would approach. And and of course, the way we count them is by the flashes that those males produced. And what we found was that males tended to produce more flashes, and as a result, be more attracted to those female flash durations that were in fact longer than the average flash duration observed in females, the flash duration, the mean plus 100 milliseconds. So again, we could imagine, if we go back, I can actually pop back for a moment to this, to see the diversity of female flash responses, with some being very, very short in duration and others being much longer duration. Again, we can see a potential selective force, at least in this one species, Phytinus ignitus, that might drive relatively long, the evolution of relatively longer flash durations in female responses. Now, this female preference and male preference for flash signals is not the only way in which these signals might be used by males and females in their interactions. Another possibility we wanted to look at, and so this is experiment 3 here, was the ways in which males might use flash signals as a way to congregate. We have in many of our fields in North America what we think of as explosive breeding assemblages, large numbers of males on a good warm summer night like we're having right now, hundreds to thousands of males all congregated together, flying and flashing. And one of the questions we want to ask were, how were males congregating in that way? Were they responding to each other's flashes? Were they responding to female flashes? So we set up similarly platforms with 4 different treatments using the virtual Firefly instruments that represented a courtship between a male and a female, a virtual courtship between one of these virtual fireflies and another virtual firefly, where they were flashing, the male was flashing and the female was responding to the male's flashes, or a lone male just spontaneously flashing, a lone female just producing spontaneously the female response flash, and then a control with nothing. And what we found there is that males were not particularly picky. They were attracted to any kind of flash activity they could see, whether it was a competition, whether it was a lone male, simulated lone female. The one thing they avoided was the platform with nothing, no flash activity going on it. It actually did have one of our virtual fireflies on it, but it was just turned off. And so males tended to visit all of those different possible setups that were flashing, using flashes as a beacon to potentially find a mate, even in cases where it was a characteristic male flash, not one of these longer duration female flashes. So. Now, this is a little bit of a detour now from North America as we move to experiment 4, because one thing we do have in North America, but I haven't had a chance to study it there, is cases in which male fireflies not only congregate, but actually synchronize their flashes as well. I haven't had a chance to study that in North America. Our most famous example of that is Phytinus carolinus in the Smoky Mountains of North America. But I have had a chance to go to Thailand and study another synchronous firefly, Pteroptex malachi. And in that case, I wanted to ask the question, was it possible that, to what extent did synchrony matter? And in what ways did synchrony matter in terms of establishing these large congregations of males on the mangrove trees along the waterways of Thailand? And so using these same virtual firefly instruments, we would set up 4 of them at a site next to the mangroves. And those four could either be synchronized at the regular Pteroptyx malachi rate of flashing, or synchronized at 1/2 the species rate, so flashing only essentially every other flash that you might normally see. In fact, here you can see it. Here's the normal rate below it. You can see it's only producing a flash every other flash, or at twice the rate. And then finally, a treatment that was asynchronous. The question was, does it matter whether it's at the species rate or not? Is the only thing that's important synchrony, in which case we would have only seen issues with the asynchronous flashes, or are these males actively congregating based on seeking out a flash pattern that is similar to their own, a flash pattern of their own species? And what we found measuring it a few different ways was that males, first off, were much more likely to approach treatment number one, which is the treatment that is the synchronous flashes that are at the species-specific pattern. They were much more likely to land at those particular, right on those virtual fireflies we'd set out there if they were flashing synchronously at the species-specific pattern. And finally, they were much more likely to actually begin to flash in synchrony with those virtual fireflies we put out there. So once again, it looks like we can look at the ways in which male responses are suggesting the importance of a particular flash characteristic. In this case, the importance of synchrony. There is, we have some reason to believe, there's a number of hypotheses for why this synchrony occurs in species like this. But certainly one of them is the beacon hypothesis that a larger cluster of males flashing synchronously might attract more females. And we can imagine that if they can establish synchrony, if they can seek out, if the males are actively seeking out other males of their own species, they can be sure to, or more likely to establish that kind of beacon. So, all right. So that concludes our thinking about and considering the ways in which males and females might be responding to flashes of their own species in ways that favor synchrony, in ways that favor large male congregations, and of course in ways that favor particular flash characteristics through preferences by males for particular female flash characteristics and preferences by females for particular male flash characteristics. But that's not the only potential evolutionary force out there shaping flash characteristics and shaping response to flashes. The other ones are predatory factors. And as I mentioned, I want to talk about two different kinds of predation. One that we have in the Americas, which are these predatory fireflies, in particular the genus Photurus, preying on other genera like Photinus. And so to examine that, once again, we set up a series of treatments because we wanted to ask the question, would Photinus predation be more of a problem if you, for instance, had a male flash and a female flash that were coming more frequently relative to one another, serving as a more frequent beacon for Photurus versus male flashes and female flashes that were more spread apart. Remember, the flash delay, that female response delay time, varies from species to species within Photinus. We also, as we mentioned, know that Photinus have single pulses, double pulses, and sometimes trains of multiple pulses. Are there advantages to having a single pulse versus a double pulse versus a triple pulse, once again, in terms of attracting these predatory Photurus? So we observed these different platforms and watched what would happen. And so when we set up those different response delays, we saw that Photurus visits as potential predators went up and up and up as you got to shorter and shorter and shorter response delays. They were using them they did seem to be using them as a cue. The shorter response delays were more attractive to Photuris, and as a result, likely would be something that would be selected against in a species that might be subject to heavy Photuris predation. We also at the same time looked at how the Photinus males themselves would respond, and they didn't really, we didn't really see a pattern in how Photinus males spent their time for this particular treatment. But interestingly enough, we did for the other treatment, where we had 1 pulse per treatment, 2, 3, or 4. And remember, Photinus ignitus males produce just 1 pulse per treatment, and they were much more likely to spend their time at those single pulse treatments than at double, triple, or quadruple pulse treatments. But once again, the Photuris were much more likely to spend their time at the 4 pulse treatments than at the less. So we can see another possible selective force that could act on flash signaling behavior in these Photinus fireflies, where if there are concerns, if Photuris predation is an issue, then producing multiple flash pulses can be a disadvantage. And in fact, some reason to believe that Photinus ignitus is actually avoiding multiple flash pulses for that very reason. And even though we know they don't produce them at all, they're also avoiding areas where they see multiple flash pulses. Finally, the last experiment I wanted to talk about was looking at a non-firefly predator, a vertebrate predator, in this case a bat. We flew bats in a chamber and recorded their flight behavior. We could get them to fly in this chamber and try to attack a little lure that we had rotating on the end of a very thin wire. And it was on the end of a wire because it had an LED at the end, either a small lure or a larger lure. And that LED was either flashing to simulate a firefly or not flashing, because we wanted to ask the question. There was a number of interesting studies that had looked at the aposematic role of glows in glowworms to deter predators. And we were asking the question, is it possible that flashes, and as a result being more active as a flasher potentially, could be advantageous in terms of deterring a potential predator like a bat? So here's our data from that particular experiment, and including another little bit of data here that just shows what these, what we call the big brown bat, Eptesicus fuscus, what the big brown bat is eating. The big brown bat is mostly a beetle specialist in North America, and so likely to attack a beetle like a firefly. We did some, it's not shown here, but we did some other experiments though, where we demonstrated, as you might expect, that the fireflies were distasteful to them. And if you tried to feed them anything that tasted like a firefly, even just a yummy mealworm with ground up firefly on top of it, or as we call firefly homogenate on it, they would spit it out and reject it. They also seemed to be much more likely to attack the non-flashing lures than the flashing lures, and more likely to attack the larger lures than the smaller lures. And here the smaller lures are labeled Photinus, and the larger lures are labeled Photuris, because those are the sizes they were meant to represent. As you can imagine, the predatory Photuris are larger, and we had seen an image of that earlier, than the smaller Photinus species. And so in this case, it does appear that flashing, particularly if you're a large firefly like a Photuris is, flashing can provide an advantage in terms of decreasing the likelihood of being attacked, at least based on what happened with these lures. So one other potential selective pressure on firefly flash signaling. Now, why is this in any way important for conservation? Well, I'm going to argue that that it's understanding these things is important for 2 reasons. The first is that as we reach out and engage in citizen science projects or community science projects, like the one that I've been involved in North America, Firefly Watch, we're going to be relying on the general public to, at least in our case, mostly be finding and counting fireflies using their flashes as a way to locate them. That's what we ask people to do. We ask them to go out and identify a firefly habitat, register online through Firefly Watch, describe the habitat, and then when they're in the habitat, note the date, time, and weather, and then do 3 10-second intervals, counting the number of flashes they've seen and inputting their data into our Firefly Watch system. And this has been interesting to get this sort of big picture image of what might or might not be happening to fireflies. But I mentioned in the outset that I wanted to talk about the potential and the pitfalls of this kind of community science work. And the pitfall is that, of course, with over 200 North American species of fireflies, if all we're getting people to do is to tell us and count the number of flashes they've seen, we're really not learning a lot about what's happening to individual species of fireflies. And so to that end, that's where it starts to become much more important that we understand the different flash patterns of fireflies, we understand their flash behavior, and that we can get the general public to be interested in their flash behavior and interested in understanding their flash behavior. And I think that's, at least for me, that's where some of these potential evolutionary stories that we've been exploring are useful as an educational tool to help prepare the public to do something more than just Firefly Watch. And I've been working on a project called Firefly Watch Pro, in which I ask folks to go out not once a week, but twice a week, still provide their location information and habitat type details like that. But now to actually tell me about the flash patterns they're seeing, so that we can begin to try to figure out what species they're observing at a particular site. So don't just tell me the total number of flashes, but the number of flash patterns, try to identify individual species by flash patterns, and so forth. And so this, I would say, is still in its infancy. We're still working on it, but in the meantime, what we're having people do is use this online system called Anecdata. It can also be used on an app on your phone to carry out these observations. And it does require some additional training. I put together videos for people to watch and to start to learn how to identify these fireflies. And while I've been doing this, in fact, the Xerces, in collaboration with IUCN, has put together an even more comprehensive set of trainings, even 3-day trainings through a project they've launched called Firefly Atlas. And so this is also a plug for Firefly Atlas for anyone from North America, but I'm sure they're going to be spreading it with IUCN's help around the world as well, as a way to once again have people go out, try to tell us what patterns of firefly signaling you're seeing, and use that working with experts to figure out what species of fireflies you're seeing. So that's, in conclusion, where I think there's a connection between flash behavior and conservation. With one final point, I think that in general, flashes are what engage people about fireflies, what fascinate them about fireflies. And so I think as we learn more about the evolution of flash behavior, more about the ways in which flash behavior is under these varying selective pressures, it really becomes an opportunity for outreach and education. And if you'd like to talk to me more about it, you can reach me here, ckratsley@fitchburgstate.edu. And I just thank all the people who helped with this, and finally put up my references. So thank you very much.
Thank you so much, Professor Kratsley, for your wonderful presentation on flash behavior. Any questions to Professor from participants, please?
Yes, I am having one.
Any questions? Sir.
Sir, I'm having one question. Can a presence of predator has any influence on firefly flash behavior?
Yes, so there's presented studies mostly that I was directly, well, exclusively that I was directly involved in, but there have been other studies that have shown the change in firefly, in particularly Photinus firefly responsiveness in the presence of Photurus behavior. So it absolutely does decrease the Photinus flash activity. And in fact, I think that's a little bit of what we're seeing in this particular figure here, where because there is a particular Photurus species that produces multiple pulses of flashes, often 4 pulses of flashes. So one interpretation of this very low amount of Photinus ignitus activity when you're producing 4 pulses of flashes is that they are in fact avoiding predators, avoiding this Photurus versicolor, a predator that produces these trains of multiple flashes.
Sir, one more questions. Like, have you found like any difference between like in the signature or pattern of firefly flash while mating or while defensing?
That's a really interesting point. So once again, if we turn to the Photurus, these large predatory species, we can definitely see a lot of examples of if they're disturbed in any way, even when you catch them and put them in a jar, they shift from from sort of a very characteristic regular rhythm of flashing to just a constant flashing that suggests to me that that's a predator avoidance flash. Similarly, what I saw in Troptic's Malachi was that as these males come flying in to join these synchronous congregations, they actually flicker as they fly in, and then they land on the leaf, or in this case on the lure that we put out for them, and then they start the synchronous species-specific flash. But as they're flying, they're flickering, and to me, that's also a potential anti-predator type of flash that they might be producing. Now, there's other interpretations, but those are 2 things I can think of that I would think of as examples of possible more anti-predator flash behavior.
Thank you so much, sir.
Yeah. Any more questions?
Enough, you don't keep your dresses properly.
Hello.
I couldn't quite hear that question.
Yeah. Please once again repeat the question. Any questions, please? Yeah. If there are no questions, we very wholeheartedly thank Professor Christopher Castley for his wonderful presentation and work on flash behavior and what it communicates to the other species or the conspecifics. Sir, thank you so much, sir, for your time and patience. And also, we would like to continue having collaboration with you. And when you visit Kerala, please do have a plan to visit Bangalore. You're always welcome to MP Bangalore, and we would be very happy to have you here as our guest.
Oh,
thank you so much. Now we from the flash behavior, we move on to yet another interesting topic that is inclusive taxonomy. So how inclusive taxonomy can help us in identification of fireflies. And this lecture is from Dr. Ishwari J. Gutierrez Carranza. He is considered a specialist in Lampyridae taxonomy. Ishwari has described one genus and 10 new species of fireflies from Mexico. He has participated in the publication of 11 scientific papers, one book chapter, and they are all about fireflies. He currently assists Professor Zaragoza Caballero as a technician curator of Coleoptera in the National Insect Collection at UNAM and collaborates with the Mexico Environment Ministry, assessing firefly population monitoring projects in urban landscapes. Very interesting. Dr. Ishwari, you may proceed with your presentation.
Thank you. Thank you. Can you hear me? Yes.
Yes, we
Okay.
can.
Now I'm sharing my screen. Can you see my screen?
Yes, we can see.
Okay. Great. Well, thank you for the introduction. Well, first of all, I'm not just speaking on behalf of myself. In fact, this is on behalf of LAF Zaragoza. For those who don't know, Professor Santiago Zaragoza Caballero is the Mexico's firefly taxonomy expert. Among many things that we do in this lab, we have several projects, but one of those, it's the Fireflies for Mexico project. In this project since 2020, we have described 94 new species from our country. This represents more or less 30% of the known Mexican fauna so far. And we also have published the first known complete life cycle of a Mexican firefly. I will show that to you later. And regarding citizen science, we have the iNaturalist platform called also Fireflies for Mexico. Here you have the QR code if you want to scan it. But anyways, I will paste the link in the chat box later if you want to take a look to some pictures of the Mexican fauna. And regarding public outreach, we also organized the Mexico Firefly Festival since 2021, which is related to World Firefly Day. Well, as many speakers already said, there are more than 2,400 species worldwide, and Mexico represents more or less the 11% of this species, with 285 so far. The most recent species was published on 1st July this year. At least in America, after Brazil, it ranks the second place with the largest richness of firefly species. Maybe the third one is USA. I don't have the updated data. Perhaps Dr. Christopher has this number updated, but well, so far, Mexico is the second place in America with species richness. I can highlight the case of the genus Photinus, because so far worldwide there are 446 species known in this genus. Naturally, it's restricted to America, but well, as Professor Roman already told us, there is one species that was introduced in Spain. But here in Mexico, only in Mexico, we have about 40% of the Photinus species. We have 177 species. Now, contrary to the Asian fauna, we don't know, well, so far, aquatic larvae they don't have gills. Basically, the larvae that we know are practically terrestrial. This is the life cycle I talked to you about. The species is Phytinus extensus. Now, as you can see in this life cycle, practically it spends like more or less 9 months as a larvae feeding on earthworms. So practically in Mexico, in most of the sighting, firefly sighting sites in Mexico, we only can see fireflies in June, July, and August, which is like the rainy season. Even in the southern places in Mexico where we have more tropical forests, even there, well, there we have fireflies throughout the year, but not the same species throughout the seasons. But well, this is the example of the pupae, the adults. Most of the females are brachypterous. Okay, as a background, it all started with the discovery of the synchronous firefly Phytinus palacios in 2012. For those who have already seen the James Cameron's movie, first one of Avatar, when they connect to this like tree and they start to make this like wave pulses, the Phytinus palacios synchrony looks something like that. Unfortunately, I don't have videos or photos, but like Professor Grassley mentioned, the International Symposium of Fireflies will be on 2025 in Mexico. So if you have any chance to come, you are invited to meet this famous firefly. Okay, so by 2021, there was an estimated record of 50 firefly sighting sites spread across 6 Mexican states. I can highlight these 3 examples where they have a huge amount of visitors. For example, Tlaxcala, where it all started, where Palacios was discovered, had a record of 120,000 visitors. I think that the first place in number of visitors sits in Taiwan with 350,000. After Taiwan, the Tlaxcala ranks the second place with the highest number of tourists. And you can make your calculations of the incomes. For example, some places have an entrance fee of $9, but most of the places from Tlaxcala have an entrance fee of 20. It can reach $21 per person. Okay. Yeah, now, each year, the number of firefly sighting sites increases exponentially. The previous were old numbers. Right now, there should be more than 100 firefly sighting sites. And most of them are managed by local communities under social property rights. This means that the sighting site belongs to all the community, and the incomes are equally distributed to the community, but they are not necessarily under regulations of the government. That's why it's called social property rights. But some of them are already registered as what so-called areas voluntarily destined to conservation, which is like a transition between social property rights towards government management, but still they belong to the community. Now, the problematic is that there's a large disparity between the number of firefly touristic sighting sites and those who actually know the identity of their species. And as Dr. Leslie Valentine said yesterday, the first step for understanding biodiversity is to know what you have. But fortunately, many communities, many local communities have the interest in knowing their diversity, their firefly diversity. That's why we created this project so-called Inclusive Taxonomy with Local Communities, which in fact is bonded to the Assess, Plan, Act IUCN species plan, with the aim of granting institutional accreditations for the firefly species ID, but also to provide talks and workshops to the communities. I will not mention all the places that we've already visited, but I want to show you that on 2021, when we started this project, we only visited one firefly sighting site. On 2022, we visited six firefly sighting sites. This year, we already visited 4. In fact, the last one we visited past Wednesday. And we hope to visit another 4 more during the mating season. Okay, so here is Mexico. And as you can see, many of the, well, most of the firefly sighting sites are located in the center of Mexico. Well, the north you can see USA, at the south the border with Guatemala and Belize. Now, if we take a closer look, this is a zoom from this look. In blue, you can see the Piedra Canteada, called Firefly sighting site, where all started with this tourism. On the red spot is the first place we visited on 2021. On green, the spots we visited last year, and in yellow, you can see the places we visited this season. So, well, here where the number 2 is located is also located the Mexico City, which is the capital from our country. And also, as you can see, it's surrounded by a chain of volcanoes. Perhaps past month you heard in the news regarding this famous volcano called Popocatépetl, which was highly active past month. But anyways, that's why this Phytinus palacios species is called the volcano slopes firefly, because it's distributed in this chain of volcanoes. And we mainly have their pine and oak forests, temperate forest. When the sighting sites ask for our assistance, we perform exploratory fieldwork visits. We also perform ethnographic work based on interviews. That's a whole another topic I could talk to you about in another talk. But basically, we also register what the community knows about their firefly species previous to our assessment. Then we capture some specimens to ID them in our lab. Finally, we generate this institutional letter for the Firefly ID. And when we deliver the letter, we give we provide workshops to the community. So, okay, so far, what have we done? Support has been provided to 11 Firefly sighting sites spread across 4 Mexican states, and we have identified 10 Firefly species, of which 2 resulted new species. We also have found 4 new Mexican state records and 1 new record for Mexico, for our country. Here is the Firefly species list that we have found common in these Firefly sighting sites. 1 species of Paractomina. Most of them are Phytinus, 1 species of Pyropegia, and finally 1 species of Phyteris. Okay, now I can emphasize the example of these 2 new species because as the project says, inclusive taxonomy with the local community. The name was provided by the local community. As you might know, Mexico was colonized, conquered by Spain during the 1500s. And after that, the official language practically turned into Spanish, okay? But before that, there was a high diversity of local languages. These days, there are 68 local languages registered in our country, and one of those languages is the called Matlatzinca language. So far, we know that there are about 50 or 60 elder people that still talk this language, but unfortunately, the younger generations have lost their ways and costumes, so they no longer speak this language. That's why the importance of this kind of dynamics with the community, because it raises awareness with the younger generation so that they can embrace again their own culture. Well, here is the example of the name. For example, the new species is called Phytinus tatiwi. In this case, to means little and tiwi means light. And that's the Matlatzinca word for firefly. One interesting and curious thing that we did in this dynamic is that the community created a neologism. A neologism, it's a new composed word that previously didn't exist in their language. As you should know, there are firefly species that when adults lack this photic organ. So we explained that to the community, and we created this word to refer a dark firefly. No, in this case, it was named Ximu. That Ximu, it was like the word referring to dark. This was a very, very beautiful dynamic with the community. Here it's a picture of when we were discussing the names of the firefly. Of course, it's not the whole community. There are only the representatives, but then they voted for the name, and well, it resulted in these 2 new species. Well, regarding the new Mexican state records, Photinus paliaciosi was, for many, many years, was believed to be restricted only to the state of Tlaxcala, where it was discovered. But after our fieldwork visits, we discovered that it was distributed in other 4 Mexican states. And regarding the new country record, we found the species called Pyractomina lucifera. So how do we make this institutional letter? We basically named the Firefly sighting site. This is highly important. We set the collecting permits. We place the Firefly species ID. We place additional comments of particularities that we see in the actual place, and of course we invite them if they have the chance to meet the National Insect Collection, because it's important for them to know where the specimens are actually guarded. Okay, and of course, finally, the signatures, which are endorsed by Professor Zaragoza Caballero. These letters are very, very useful to the community because when they want to register their Firefly sighting site officially to the government, they need to know which species are they trying to preserve. Here are some pictures when we delivered the institutional letter and when we delivered the workshops to the tourist guides mainly and the representatives from the community. We also try to provide them public outreach materials. Here is an example of 2 scientific dissemination posters, in which the information is restricted to the Firefly sighting sites. Of course, we deliver them the digital documents so that they can print as many copies as they want and spread them through their Firefly sighting site. This is one scientific paper that we published regarding the exploratory visits, the distribution of Photinus paliaciosi. The 2 new species that I talked to you about from the Matlatzinca community is now fortunately in press. Hopefully it will came out this year. And regarding the Pyractomina lucifera record, we need to confirm if it's really this species, because so far it's known from USA. I'm not completely sure if it's also registered in Canada, but if it's not Pyractomina lucifera, it should be probably a new species as well. Well, future work, yeah. Well, unfortunately, not everything is like a fairy tale. It's a common problem, right? The light pollution and pesticides and everything. So we still need to do more public outreach work. Fortunately, this project was selected by the IUCN Firefly Specialist Group committee this year to be submitted to the IUCN grants. So we have we hopefully hope to win this year to get fundings, because like Professor Sergio Enriquez mentioned, unfortunately, this work is not possible without fundings, right? Also local stakeholders, but mainly funding. So we hope to get this grant this year so we can visit Firefly sighting sites the next the next year. Well, finally, I would like to thank, of course, all the members of Lab Zaragoza, mainly Dr. Edwin Dominguez, who is also part of the IUCN Firefly Specialist Group. And he is the person who is almost always with me on these exploratory visits to the sighting sites. And of course, to all the places that extend the invitation for us to visit their and know their fireflies. And of course, Embry and Dr. Chakravanti for the invitation. Finally, if there are any questions and if we still have time, if no, here is my contact info, my email, and here is the QR code of the Lab Zaragoza social media, where we usually publish like what we're doing with the communities. And that will be all. Thank you.
Thank you, Dr. Ishwari Gutierrez, for an overview of your work in Mexico. And now we invite any questions from the audience. Any questions to Dr. Ishwari from participants? Yeah, if there are no questions to Dr. Any questions, Madam Leslie Valentine? Any questions?
I just wanted to make a comment, Ishwari, that I'm green with envy. You've got a whole lab behind you over there, and I've got only me. So all of Australian firefly work just sort of falls on my head. It would be wonderful to have the sort of situation you have, and I think it's just marvelous what you've been able to achieve over there. So I'm as I said, I'm very envious.
Okay.
Thank you, Dr. Leslie. Thank you.
Yeah. So this has been a very interesting lecture, and I hope that the participants will have many questions, and I'm sure they will be contacting you in due course of time, Dr. Ishwari. So thank you so much for your presentation, and which was very and highly informative. Thank you, sir.
Thank you. Bye-bye.
Yeah. From the Mexico, we move on to China. We have with us Miss Li Ping from China Biodiversity Conservation and Green Development Foundation, China. Ms. Ping Li, Director of Community Conservation Area of China for Biodiversity Conservation and Green Development, has been focusing on environmental issues since 2014. She is committed to research on environmental pollution and started to preside over the protection of fireflies in 2016. She has presided over projects such as Firefly Ecological Line, Keeping Fireflies at Home, and is currently focusing on the protection of fireflies in Guangzhou, initiating the activity of Disappearing Firefly Park. She is currently focusing on the protection of fireflies in Guangzhou and initiated the activity of Disappearing Firefly Park. Ms. Li Ping, please.
Is anybody see my screen?
Yeah.
Yes.
Yes, we are hearing you, and we can also see your screen.
Okay. Hi, everyone. I'm Ping Lei. I'm from China. Today, I will introduce our action Disappearing Firefly Park in Guangzhou. And now we can see it is a highway expansion project. And equal VS firefly habitat protection with a total investment of 15.7 billion Shenyang Haikong National Highway Expansion Project, the first expansion project of Guangzhou North Second Ring Road, has started in December 2022. The disappearing Fireflies Park is on the other ring road, other side, the same project. And the expansion areas of Banzhou interchange involves Tianluhu Lake National Secondary Forest Park, which will be expanded from 6 lanes to 12 lanes, as well as the construction of the bridge on both sides of the highway. And its expansion area highly overlaps with the firefly habitat, which is a rare ecological habitat in Guangzhou with 10 firefly species at least. Now we can see the picture. In this picture, we can see within 80 meters on the right of the highway, 10 species of fireflies are distributed in several places. We want more people know what will we pay for the runway expanding and consider if it was. Now, and then a disappearing firefly appear. Now we have a look at the video.
Wait. I mute her. Ms. Pingley, can you hear us? Are you playing a video? We are not able to see it.
Internet lost.
Hello, if you can hear us, you can unmute yourself and respond. If you were showing a video, we are not able to see it.
Now, if anyone can see the video.
No, we are not able to see the video.
Oh, I am try again.
Yeah, can you just share your screen again, please?
Yes.
Okay.
If anyone can see the video.
你这手动对讲啊？真麻烦。OK。
Hello, Pingley, you can try again. You're muted. You can unmute yourself. Miss Ping, yeah, can you unmute yourself? Yes. Can you try sharing your video again, please?
Okay. Hello.
Hello, thank you. That was a lovely video. Could you just go on full screen mode? There's an arrow on the top. There's an if you could go on full screen, please. There's an arrow on the top of your toolbar. If you could just make that, because we can see it in your presentation. The one next to it on the left-hand side. Yeah, the one, yeah, that one. If you can just.
Oh, okay.
Yeah, that arrow. Yes, that one, not that one. Can you just get play button in the present button?
And can we find yes. Yes. Yes, it's okay.
Yes, please go ahead.
Yes, anyone can see it.
Yes, please go ahead.
Yeah, please go ahead. Please go ahead.
Okay. Did you say the video and okay.
Yes, you can start.
Have anyone can see? Can watch the picture?
Yes, yes, yes. We can
Yes,
see your
okay.
PPT.
It is not a common city park. This Disappearing Firefly Park is a wildlife paradise composed of group of Homo sapiens, Guangzhou citizens holding hands with fireflies, spiders, walk. Hands with firefly spiders, frogs, snakes, butterflies, and other population. The park is donating the brightest fireflies at night. It officially have opened at 1930 on July 14, 2023. And then we also hold night tours from time to time and encourage the public to visit. Where it is, it's just locating in the corner of the Guangzhou Tianlu Lake Provisional Forest Park, about 2 kilometers away from the Guangzhou open area. And now we can see the guy have take photo with me. In June, I came here and investigating the firefly frequently with my partner. We found just has 10 firefly species were found in this little place, include an unknown larva full body glow. Now we can see the first. Now we can see the picture. Different firefly species on here. Now we can see the first census. It's an orange point surrounded by several kinds of fireflies, such as the guy and Antiquata lay. And
can you not move to the next slide?
Yes, it's a Cantica lay.
Can you move to the next slide, please? We can't see the slide that you're presenting.
What? Can you speak loudly? Now, can anyone can see?
Yes, you please proceed, Ms. Ping. Yeah, you proceed.
Now we can see the Cantica lay. It's in the orange. It's an orange point surrounded by several kinds of fireflies, such as this guy. And then the larvas live in the water. And we can see the Rancilla lunata is this guy live on the wet soil by this pond. Next to the pond is dense bamboo. The Stephanus Cuternus, seconded in small screw under the tall miscanthus by the pond. The water gave birth to a contact lay, and for this one, the humid air environment created by the pond is very important. If the ponds disappear, will they still be there? And now we see the second sense, feng shui forest. Feng shui forest is intentionally designed and arranged according to the principles of feng shui. The special forest is gently built in vulnerable location with the belief that they will attract positive energy and bring good luck and blessings to those who inhabit the area. The forest will already disappear for the highway expansion. It's buried. Now, the forest leaf and Luciana Curtis Oxica. 2 months ago, I took photo of Luciluna succiorisa here. Their members lies out the forest night. Now no one knows their praying, their living habitat. But this one, the larva be raised. And now we have been trying to figure it out, but haven't got anything. And we can enjoy the picture of this one. Their habitat. And the third sense is the Cantus vercampitinis. In the valley of feng shui forest, we can enjoy another species, like Cantus vercampitinis. Let's enjoy the habitat. Yeah, their home. Back to this Bering firefly park, there is also lots of another species. Let's can see. To protect several firefly species, we also took action to add extortion. It's a light invasion. Light invasion is a field that a black fishing net creates. It's the night for fireflies. Everyone takes away a lamp when they leave. The presence of the light getting darker symbolizing the disappearing fireflies due to human activities. Okay, let's enjoy. It's not only the firefly habitat is always have another life in here. The frog, butterflies. We know in addition, firefly, we know that they have many interests live here. So we don't want but we don't know the future of the firefly habitats and if it could be researched, but we need have a try. So it's the reason that we built the Disappearing Firefly Park. We hope more people know about the Disappearing Firefly Park before it is covered by highway expanding architecture. Nowadays, more and more people come here and forbid farewell to the firefly flock plants by themselves. Also, we appeal the government to consider keeping firefly habitat. Lots of people come here and they're very sad about to hear the news. Now there are only 41 days left for the highway expansion to begin. We need your help for the habitats of the several fireflies. For example, support by making your voice and discuss more feasible solution with us to keep their habitats. And you can contact me, Ping Lei. Thank you, everyone.
Yeah. Thank you very much, Lee. Any questions to the speaker, please, from participants? Any questions to Dr. Lee Pink? Yeah. What's the question? Yeah, there's one question from Nishant. Do you happen to know an estimate of the number of firefly species in China? How many species of fireflies are there in China? Any estimate?
In China, they have 130 in the species.
More than 100 species, is it?
130, I think so.
Yeah.
And a lot of
more than 100 species.
Yes, more than the.
You have chance. Yeah. Yeah, Professor Leslie Valentine has some comments to make.
Yeah, well, Oliver Keller is going to be speaking later on, Ping Li, and he will be able to tell you exactly how many species there are in China. So, but the other thing I wanted to ask you, who identified those fireflies for you? Do you know?
Huh?
Who told you they were Pyrocelia and Aquatica lei?
这个地方的是谁画？What are the ways to showcase for future studies? Wait a minute.
Well, listen, can I was just going to suggest a name to you. It wasn't Professor Xinhua Fu by any chance, was it?
Oh yes, I heard about him.
Good. Good. That's all right.
And another question.
But another person was probably me.
Yeah. Yeah. Any more questions to the speaker from China? If there are no questions, definitely you can contact her. So we move on to Malaysia from China. Thank you so much, Li Ping, for your presentation. Okay, and I hope you will continue to work for firefly conservation in China and be in touch with us. Then we move on to Malaysia, and we have with us Miss Veronica Koo. Miss Veronica has Bachelor of Science in Zoology and Master of Science in Applied Entomology from University Science Malaysia, and she's from Penang, Malaysia. She has been working on congregating fireflies for the past 16 years, focusing on firefly conservation via a long-term monitoring program conducted in a popular firefly tourism destinations in Malaysia. This program has been carried out longer than her work experience. Besides the monitoring program, she has expanded her studies to ecological and behavioral aspects, looking into the different rivers and estuarine areas in peninsular Malaysia. She has been also studying impacts of artificial light on the congregating fireflies. Through the years of firefly studies, various technical and semi-technical publications have been published, and one notable publication has been Conservation Action Plan for the Fireflies of Selangor River, Malaysia. This publication is hoped to be able to serve as a reference for local stakeholders and for implementing agencies. So, Dr. Veronica Koo, please. Yeah, she's not audible.
Sorry. Can you hear me now? Can you hear me?
Yes, ma'am. Yes, ma'am.
Yeah. All right. Okay. Sure. Thank you. Thank you, Dr. Chakra, and thank you everybody for the invitation. As mentioned, my name is Veronica. So we are here today to share about what we have done so far in monitoring congregation fireflies, and we are using a non-destructive method, which is known as digital night photography. So just a little sharing. Okay, so just to show you slightly, so in Malaysia, we have two types of fireflies. One are known as solitary fireflies, and the other one is the congregating fireflies. So just to show you an example, so the congregating fireflies looks like a Christmas tree from the riverside. So some of the questions that we often ponder are actually, what is the status of the firefly population? I think globally, all of us want to know, like, how is our population doing? And also, what are the threats to the firefly population? I'm sure all of us are we are very used to hearing about threats to the firefly population, being habitat loss like pollution or any chemical usage. And also, we know that there are threats. So what are the possible remedial actions to be taken? Either rehabilitation, minimizing the usage of lights, or even more research being done on fireflies. So how do we address these questions? We actually can do so by determining the population trends. So by doing so, we need a systematic monitoring program for a long-term basis. And we are very happy to share today with you some of the methods that we have done so far. So by having a systematic monitoring program, we can actually assess what are the possible effects of environmental changes on the firefly population. And with the findings that we have, we can actually help to inform the stakeholders or our government on what are the implementation of measures and actions that can be taken to sustain the firefly population. Just briefly, we know that we have been doing various studies, and we always involve sweeping nets to collect them, sticky traps, which is actually not recommended. Visual scores are actually those that we count on a tree, and then and we range them from low, medium to high. But when there's more than 100 individuals on one tree, the sensitivity is actually lost. So it's not a good way to use as a method to conduct monitoring. So one of the methods that we actually tried out also is actually to use a cardboard, and then we cut out a little window frame, we measure it, and then from the river, we actually have like a distance from the riverbank, and we use that to count the number of individual fireflies in that particular window. So the person has to be actually a very trained observer to handle this counts. And usually it has to be either the same person or at least one or two that has been calibrated among them. Besides using that, there are also methods such as using photo multipliers, but these are very expensive equipments, and it's very hard for us to bring them to the field. So it's not so good a tool to use to measure the firefly abundance. So next, or the final one that we actually thought about is how about using digital night photography? So we actually embarked on this study, and we had numerous trials on using the techniques, having to fine tune the method as well. And finally, in 2012, the method was actually published in Insect Conservation and Diversity. You can, for further information, you can actually search this paper and you can read through it to have a better know. But for this particular session, I'm just sharing with you what we did to come up with the method. So for the method actually involves using photography. So we have a camera. Back then, the latest model was a Canon 5D. And because we had some additional funding, so we managed to get an upgrade of a Canon 5D Mark II as a backup. So for the lens, we actually used a telephoto lens with the MIG from Canon as well, ranging to 200 mm aperture of 2.8, and then with image stabilizer. With all these camera bodies and lenses, they're quite heavy, so we require a very sturdy tripod to place them. So if you can see the picture on the left, is actually to show you there's a tripod located in the mangrove area, because we are actually photographing across the riverbank. We actually depend on the dial, tripod dial, where you can see the angles. So because we are doing monitoring, so we have to be consistently on the same angles. So we use that as an indicator. But because of, sometimes the dial having some problems, so we decided to use a compass. So we modified a compass holder to place it on top of our camera. So you can see at the picture on your bottom right. So just to share with you, our monitoring sites, we actually have 7 of them located, spread out across the Selangor River. And each of this site is actually, how we chose them is, first we have to go by the boat to see what sites are best for us to carry out photography. And when we have already determined the sites, we have to actually go across the riverbank to see whether there are places that are suitable for us to place the tripods. So when this is possible, especially we need to go by land, because if by water, it will be hard because you have the tide levels, and if low tide, you can't get up to the riverbank. So there's a lot of considerations that we had to take in before we come up with these 7 localities. Just to share with you also what goes around behind the scenes of how we come up with our work. So on a monthly basis, we do the monitoring. We do actually do monitoring on snails as well to see how is the food source for the firefly larvae, which is actually adjacent to the site, one of our adult sites. So we have our team of people doing surveys during the day, and then also we have to do some pruning. If you could see just a short video, our team have to carefully climb the tree to do some pruning, because we don't want those foliage to actually block our camera view, but we do not cut the whole tree down. We just do minimal pruning of the trees in order for us to photograph trees across the riverbank. So, and on some nights, we could also see some bioluminescent fungi during our trips. How do we set up our tripod and do the photography? As mentioned, we actually have 7 localities throughout the river. So we carried out across 3 days, and we usually do it around new moon sessions. And in fact, we actually just came back from our fieldwork on Friday. So we have these fixed locations, and we use ground markers. We have placed permanent ground markers in each of the site. And the tripod is actually being set up using a plumb line as a reference, so that we know exactly where is the position for us to place every month. But during the day, we actually do pre-focus our camera first, so that during the night, we can actually use manual settings. And for focal length, it's actually prefixed for each of the respective sites. For ISO and aperture settings, we are actually using the highest ISO possible for the camera settings. Aperture is the lowest number possible that we could have. For our case, it will be 2.8, and ISO sets at 3200. So what we do is the images are actually taken along a stretch of display trees, and we have a start and end markers placed across the riverbank for us to know where is exactly the site that we are supposed to monitor. So we do this across every night for respective sites, and each of the site we actually have replicates, 3 replicates of the images taken. We take at short exposures of 0.5 seconds for us to get the data, but we do have longer exposures for us to use as reference to see where are the images that we're supposed to crop. Just to give you an idea, we actually do photography across the riverbank. So you can see that the camera is on one side of the riverbank, and you actually shoot across at night. So the fireflies will be actually displaying along that stretch of display trees. It's known as mangrove apple trees, or Soneratia cassiolaris. So after we have gone to the field, we will come back. Those images are renamed according to the sites. And then what we do is we use Photoshop to crop the images, the overlapping images, where we have to enhance them and then crop where there are some overlapping, so that we get a full stretch of it without having bias in our counts. So after having done that, we go through the analyzing of the images, where we use a software called analysis software. It's actually made by Olympus, but it's no longer in production. So because it's only supported until Windows XP, and we know now that there's no more Windows XP available, but we can still use this because our computers, some of our computers are still functioning on Windows XP. So what I can share is that previously, the team from Olympus, they helped us prepare some macros for the software, and it is able to detect the spots on the firefly, on the images, and then give us a count of the number of fireflies present in that particular frame. But on cases where there's too much noise, we could actually do it manually. So by doing so, we have to enhance the images. And if you could see on your top right, those little spots that you see, and we use the software manually to count them. And after you finish it, you can actually, they will tabulate the count for you at an Excel sheet. It's a spread out sheet. So for the case of now, because this software is no longer in production, I'd like to suggest that some other potential software that could be used is ImageJ or Fiji. They are almost alike, but from my experience, I think Fiji is the one that can also give you some the tabulations right after you finish your counts. For this .goose, I came across it, but I cannot use it because we don't have Mac in our office. So I think for those who are using Mac, maybe you could try out and maybe share with us our experience, whether you could use it as a count, because I understand that it's actually developed for the purpose of counting object in images. So I think it would be a good one. It's just that we don't have the Mac in our office to use it. So we can't be able to try it out. So just to share with you some of our results and findings through the years that we have conducted the monitoring. As you see, across 16 years of data that we have, initially there's a shift in population trend, where there are certain months, those earlier of our monitoring sessions, you could see that they peak in the middle of the year, but this shifted to end of the year, and then it went to early of the year, and then now it shifted back to middle of the year. But the peaks are no longer that obvious, and the whole possible idea for us about the overall shift, it could be because of change in rainfall patterns that we are experiencing. Those days we are quite fixed, where you know that this particular month you have heavy rainfall, but these days you don't get that occasionally. You have rainfall in different months is unpredictable anymore, you know. So besides the shift in rainfall, the shift because of rainfall patterns, other factors that are having a problem, giving an effect towards the firefly population are habitat loss. We do have sometimes unusual high tide phenomenon where there is temporary inundation at the sites behind the display trees, and these are where the firefly larvae are found, and the snails. And also recently in 2020, we had quite a bad flood in that area. And also in 2023, there was slightly another flooding in late of 2022, and there were a few trees, display trees that died. So there are some other possible factors that are giving effects towards the firefly population. Some of the findings throughout the years that we had was, as I mentioned earlier, that we are actually doing it on a monthly basis. So habitat loss was detected because we could see the display trees in front, but we cannot see the habitat behind it. So all these were reported to the local authorities and the government. And the area is now gazetted as a protection zone under an enactment from Selangor Waters Management Authority. And also the state had reacquired certain sections of the land for rehabilitation. So you can see the pictures below. The area was a degraded area, and then in 2 to 3 years, there was a regrowth, and right now it's left to grow as well as possible to try to create a natural habitat again. It's not easy doing it because a lot of work was involved to try to get the habitat to its original, not say original, but to how it can be at that very moment. You know, a lot of awareness has to be given to the public to not clear the land entirely. So some of the problems that we encounter throughout the years of monitoring is one thing, which is the major problem for us would be erosion. If you could see, because we are always right at the river bank, and you know, rivers, they constantly have eroded river banks, and all our sites, monitoring sites, are actually located on the concave area where it's more prone to erosion. So we are constantly having to shift our sites slightly behind, but still we are able to do our monitoring for 16 years and counting. So before I end my presentation, I would just like to thank Dr. Chakra and his team and EMPRI, also the funders for the study, which is actually Department of Irrigation and Drainage Malaysia plus Danida, Selangor government, and also not forgetting our team at entomology. Without them, we won't be able to get all this data through this years. With that, I thank you.
Kaku, for your very nice and interesting presentation. Any questions to the speaker, please? The study has been very complex, and it's very difficult to dissect the aspects in different sections and then make up the study and estimate the population of congregating fireflies. Any questions for the speaker, please? Okay, if there are no questions, again, we thank Dr. Veronica Khoo from Malaysia for her very interesting study, and definitely I hope she will continue to unravel the full aspects comprehensively on congregating populations of fireflies. Thank you so much. We move on to next speaker, Professor Jeremy Niven from University of Sussex, United Kingdom. As an undergraduate, Professor Jeremy reached UK. He did his PhD in neuroscience on limb control in locusts. He subsequently worked on vision in fruit flies and spent time at the Smithsonian Tropical Cambridge for a Royal Society University Research Fellowship. He moved to the University of Sussex in 2012, where he is currently Professor of Zoology at the University of Sussex, UK, and also the Dean of the Doctoral School. His research focuses primarily on insect behavior, physiology, neuroscience, working on a range of species such as locusts, termites, wood ants, bush crickets, and of course, the last but not the least, the glowworms. Professor Jeremy Niven, please.
Can you hear me?
Yes, we can hear you, sir.
Great. Okay, thank you very much for inviting me to talk to you today. I think in comparison to some of the talks that we've just heard, mine will be more limited in the sense that I'm going to be talking about just one species, the common glowworm, in just one location, which is the UK. Just for those of you who aren't familiar, on your left on your screens, you can see a male glowworm. He's just walking up a stem of grass and about to take off early in the evening. And you can see a female on your right. Her abdomen is lifted upwards to reveal her glow. That's the kind of yellowy area that you can see there. I'm going to be focusing on common glowworms. They're members of the genus Lampyris and the family Lampyridae. As we've heard in an earlier talk, adult female glowworms are larviform. In a sense, they retain a larval body structure and they lack wings. They're sedentary and they show a great deal of site fidelity throughout the season, so that females that aren't mated on any particular night will be found in almost an identical location on subsequent nights, still glowing. And females use that glow to attract the volant males. They fly in to the females, and typically males don't land exactly on the females. They'll land slightly in a nearby area, and they will then complete their journey to the female by walking to her. And here you can see a little video. Here's a male who's found a female, and he's attempting to initiate sperm transfer. And what you can see is that the female remains glowing throughout that period. The male's probing her abdomen and attempting to initiate a mating. So common glowworms are, at least according to Tyler, spread throughout Europe. They're very widely spread. And as we've heard, they're certainly present in large tracts of Southern Europe as well as Northern Europe. And they're also present in Northern Asia, all the way across into Northern China, large areas of Russia. The populations that I study are centered in Southern England, and you can see on the right-hand map, a map of Southern England, and it's just plotting out some of the various different sightings. Each one of those red dots is a reported sighting of a female glowworm. And at the moment in our database, we have around 16,000 sightings of female glowworms, though very few sightings indeed of males, which are much harder to locate for members of the public. So one of the reasons that I started working on glowworms is because of a report, quite a recent one, by Gardner and Didem, which suggested that glowworms in the UK were at least going locally extinct, so populations were disappearing where they had formerly had strongholds, and that this might be linked to artificial lighting at night. And within the UK, at least, we're able to plot these various different data points for individual female sightings and overlay them with a satellite imagery of artificial lighting at night. And hopefully what you can see in the map that's on the right-hand side of your screen is a lot of small green dots. Each one of those is a reported sighting of a female, and they overlay the map of artificial lighting at night. The UK is very densely populated and has a great deal of artificial lighting at night, and so there's a very strong overlap between the presence of females and the presence of artificial lighting at night. So it's obviously of a concern if we think that artificial lighting at night is actually affecting the longevity and health of populations. Before I get into any of the specific experiments that I'm going to talk about, I just wanted to mention one thing, which is the strategy that we use in order to study male glowworms. So we generally use funnel traps that we can place out in the field. These are very simply made from plastic bottles, and they contain a green LED. And at the bottom of your screen, you can see a small illustration of one of those traps. And these funnel traps often catch many thousands of males in a single night. The LED that we use is matched to the emission spectrum of the female glow. So there's a lot of correspondence between them. It's approximately a 550 nanometer female glow, and the LED matches that. So what we wanted to try and understand is what's going on for the glowworms in terms of the effects of artificial lighting at night. And we were particularly interested in the effects on male glowworms. They're, as I said, they are the major dispersers within this species. And so they are much more likely to encounter artificial lighting at night. We also know that females of this species typically do not move away from artificial lighting at night if they are exposed to it. So males, in order to get to them, have to go through that lighting. In order to kind of assess and quantify the effects of artificial lighting at night on males' ability to find females, what we did was to construct a transect in the field in an area where we knew that there were glowworms present, and it's part of a dark skies site, so that there's minimal artificial lighting around. We had a series of traps, 40 in total, and in the middle of those traps we had a single white light, a white LED light that mimicked the emission spectrum of street lamps, and you can see that on your bottom, on the bottom left-hand corner of the screen. And we had 10 traps covering the direction that was illuminated by light, and we had a matching set of traps that were in the darkness, so a matched control. What you can see on the right-hand side of your screen is a graph of the comparison of the numbers of males that were caught in traps, either if they were illuminated or if they were in the dark. And hopefully what you can see is that traps that are very close to the source of white illumination are very poor at catching males, and so those are shown in the yellow, whilst the equivalent traps at the equivalent distance from the light source but in the dark are much more effective at catching males. And indeed, there's about a 95% reduction of those of male capture due to the artificial lighting, and it's highly significant. So that gave us an indication that artificial lighting is extremely important and can be highly deleterious in terms of the ability of males to reach females in this species. But what we didn't know is exactly what the effects were of the artificial lighting itself. Was it affecting the ability to detect females? Was it affecting the ability of males to actually perceive contrast within the system? And so we took males back into the lab and placed them into a Y maze. This Y maze contained a glowing LED in one of the arms, and we looked at the ability of males to reach females. And I've just, if I can activate the video, I'm just going to play for you a short video that shows the males in action in the Y maze. So here you can see a male, that one of the arms is illuminated and the male walks off to the green LED. where he sits and looks rather confused because there's no female there. We can also then increase the amount of lighting. So this is with a 25 lux white LED white light source. And here you can see the male is far more reluctant to move. But eventually he does move, and he moves in the correct direction. Though notice that he remains in the shadow of the Y maze as he moves along the arm. And there he goes. And then we were also able to increase the level of lighting. And so what you'll see here is a 90 lux light source, so far, far brighter. And here the male is hardly moving at all, and has a great deal of difficulty. I'm not going to play either. So what we did then was to analyze the data, and we could do that in multiple different ways. Initially, we looked at the ability of males to find females, and we placed the males into the Y maze multiple times and looked at their ability to locate the female. And what we discovered is that the males, even if they're in the darkness, they will start to tire or perhaps lose interest, we're not sure which, in terms of the females. And so as they spend more time in the Y maze, they're less likely to move towards the dummy target. And this should perhaps make us think about exactly how males are perceiving the female and the amount of flexibility they have in controlling their behavior. And that's highly significant as a result. And I'm showing you here the logistic regression. So males that reach the female are at position 1, and males that fail to reach a female are shown at lower as position 0. We can do the same thing with a Y maze that's artificially illuminated with white light. And so here what I'm showing you is a similar sort of logistic regression, but where the x-axis is now light intensity, so the different intensities that the males were exposed to. Again, you can see whether the males reached the female, which is shown in 1, or whether the males failed to reach a female, which is shown at 0. And again, this is highly significant, suggesting that artificial lighting and the strength of artificial lighting substantially reduces the ability of males to find females, and does so in relation to the intensity of the white lighting. Having males in a Y maze enabled us to be able to look at their behavior in far more detail. And so we could consider a whole range of different behavioral measures of how the males were performing in relation to getting to the females. And here I just want to emphasize one of them, one of the several that we took. In this case, we're looking at the ability of males to retract their head beneath their head shield, and therefore shelter their eyes from the light, or extend their eyes forwards, which is what they typically do when they're searching for females. And in the dark Y maze, there is a very small but significant shift in the latency for males to emerge with their eyes from beneath the head shield at the beginning of each trial. However, what we see with the illuminated Y maze, where there's artificial lighting, is that males retain their head shield, retain their tucked in position beneath the head shield far longer. And in fact, in many cases, males do not move their eyes forwards out of the head shield in order to search for females. And so they're remaining quite limited in terms of their visual abilities. We incorporated several of these different behavioral measures together, and what you can actually see is that they largely correlate with one another, and there's a whole suite of different behavioral changes that are happening to males when they're exposed to artificial lighting, and the GLM that we used here was highly significant, suggesting that there is a very, very strong prediction of the data. Of course, the Y maze itself doesn't tell us exactly what's happening in the field. And in order to interrogate what was happening, what we did was to go back out to these dark sites where we know glowworms are present, and we erected a large lighting feature. In this case, it's a 30-meter transect of white LED lights that mimic street lamps. They're placed onto tripods and they cross the entire site. And what we wanted to do was to test whether this sort of lighting, when it's not directly affecting or not directly over the females, but is simply in proximity to the females, how does that affect the male's ability to find the females? And specifically what we did was to adopt a strategy using a mark release recapture. So we captured a large number of males, we marked them, and we released those males at the site in darkness, and we just looked to see whether the males would remain where they were after marking until the next day when we were able to do the experiments. And what we discover is that males remain where you release them largely. And so that was very important for us in terms of the way we designed the next experiments. And here on your right of your screen, you can see a graph which just shows you the distance from the release site and the number of marked males that we recaptured on the next night. And you can see that almost all of the males that are recaptured within 5 meters of the original release site. And that suggests that when we mark males and release them, they remain where they are until the next night. And we're going to use that in the subsequent experiment, which I'm going to show you here. So we had a set of traps which mimic females. And we did that for a control, which had a control release point. The set of traps were 30 meters away from the control. Or we did that for a test release site, which is where the set of traps were 30 meters away from a release site, which had interposed between the release site and the traps a barrier of white light, as I've described, this 30-meter long white barrier. And at the control site, we released 1,199 males, and we released a similar number at the test side, where the light barrier was present. And we looked to see how many of those males were able to reach the traps nearest them, and also the other traps that were present in the experiment. And these are always matched experiments. The controls are done at the same time as the light barrier tests. And what we discovered is that there is a very, very strong effect of the presence of an interposed light barrier between males and females on the ability of the males to find the females. And what you can see here on the right of the screen is the number of glowworms that were discovered at the traps and the distance from the release site. And we can see that in the presence of so in the darkness, so for the control animals, a very large number of males were found, well over 140 or something, males were found in the 30-meter traps. And a smaller number were found in the other traps that were present at 80 meters. Conversely, when we looked at the traps nearest the light barrier, what we discovered is that in the 30-meter traps, there are far, far fewer males discovered, and that in the 80-meter traps, there are also a relatively small number of males discovered. And in fact, the effect size of the light barrier is an 82% reduction in the number of males that are caught as a consequence of the presence of the light barrier. And there is no significant effect in any of the other traps at all. So in conclusion, what I just wanted to kind of emphasize were some of the things that I've mentioned throughout the talk. So in the field, direct white artificial lighting at night applied to dummy females can certainly prevent males from finding them. And this effect increases with proximity to the light source, so the intensity of the light source. In the laboratory, white light illumination of a Y maze prevents males from reaching females and increases the time that they take to get there and elicits a whole range of different behaviors. So they really are trying to mitigate the effects, but equally that the lighting is still having a very big effect on the time it takes to reach females and on the number of males that eventually get there. And finally, that a linear lighting feature, which mimics street lighting, certainly in terms of its spectral emissions and certainly in terms of its extent, impairs the ability of males to reach females and effectively acts like a barrier within the environment to stop male movements to reach females. And with that, I just wanted to conclude and thank the people who did all the work with me. So Dr. Estelle Mubarak, Dr. Sophia David Fernandez and Dr. Craig Pearl were all working with me, and Professor Alan Stewart, who's been a long-term collaborator. And I wanted to thank my funding sources, the BBSRC in the UK and the University of Sussex. Thank you very much.
Thank you so much, Dr. Joey. Any questions from participants? Yeah. Sir, you have one question. Is it only light that make males search for females, or is there any possibility of the role of pheromones too seen in mating process of fireflies?
I can't speak specifically for the species that we work on. We use light in order to attract the males, and certainly the males are attracted to the light. But given the way that they walk to females and the environment in which they're walking through, the very, very cluttered environment, it wouldn't be surprising if they also were combining local pheromone signals with the lights that they're using to create longer-term, long-distance attraction. So that's certainly possible.
Yeah.
I have a question.
Yeah.
I saw that you varied the density of the light. So have you tried to put like a LED that simulates the female on one end, but then also other LEDs that have different frequencies to simulate having a female, but also light pollution, and if the male can still pick up the female despite the light pollution at different densities?
We have done that experiment, and in fact, what we discover is that certain wavelengths, particularly very short wavelengths, are extremely effective in disrupting the ability of males to move towards females. So they're very, very sensitive to any short wavelength emissions that are nearby the female. So what we do is we pair two LEDs, one that mimics the female and one that is a short light emitter next to one another. And when you do that, the males will no longer go towards the female. So they're very,
looking forward to your papers. I just found the one that you mentioned, and looking forward to the other one.
Okay, thank you very much.
Dr. Urjit has one question. Have you ever experienced the impact of increased temperature and warming events that England has seen in recent times on the mate finding behavior of fireflies besides the artificial lighting?
Yes, certainly. There are very, very substantial effects of temperature and rainfall, particularly in certain periods of the year, which appear to be, I can't be sure of this, but I think close to the final larval instar and the emergence from the pupa. They seem to have huge effects on population sizes. And we have a 20-year-long data set that we're analyzing at the moment that suggests that there are very, very substantial effects of both temperature and of rainfall on glowworm populations, beside the effects of artificial lighting.
Questions to sir? Yeah, if there are no questions to Professor Jeremy Niven of University of Sussex, UK. Sir, your presentation was very interesting. and it dealt with artificial lighting and behavior of fireflies. Of course, it is compounded with other factors, maybe because of pheromones or heat waves or temperature rise or rainfall pattern. Okay, so that's has been a very interesting presentation from the UK. We thank you immensely, Professor Jeremy Niven, for your wonderful presentation, and hope you will be in touch with us. With this, we have a short break of 1 or 2 minutes, and an announcement is there. After that, we will have Dr. Oliver Keller from Florida Museum, USA. So may I have the brochure, please? Just a minute, and then we will move on to Dr. Oliver Keller. We have one announcement on the brochure. Yeah, so this is the brochure we have released for the common people in India. This has been published both in the local language, Kannada, and also in English. So we have published a number of such brochures, and they are available with us, and anyone interested can access them or have them. So this is one of the brochures we have released, and we're also given to IUCN for its dissemination. And then, dear participants, we have one Asia Pacific network. We have established this network for the firefly workers around the region of Asia Pacific, including Southeast Asia, to exchange and also to have a platform for solving some of their issues or having some queries. So we have this Asia network, and many of the workers from the Southeast Asian countries are members of this. So this is a very fertile platform, and definitely the workers, especially the young ones, are encouraged to log on to this Firefly of Asia Pacific network for more information and for sharing the experience and experience. Ladies and gentlemen, now we move on to the last lecture, but not the least. Very important, Dr. Oliver Keller from Florida Museum, USA. Dr. Keller is a biological scientist for the Florida Department of Agriculture and Consumer Services. He works in the Florida State Collection of Arthropods. He received his PhD from the University of Florida, working on the fireflies of the West Indies, especially from the Hispaniola. He currently works on firefly diversity in Belize, El Salvador, Guatemala, and the Honduras, and an update of the catalog of world fireflies. A very challenging and daunting task indeed. Dr. Oliver Keller, please.
Hello. Let me share my screen. There you go. Share. All right. Can everyone see the screen and hear me?
Yeah, we can see. Yes, please go ahead.
Okay. So first of all, thank you for inviting me to give this talk. I have to say that when you did, it was a little daunting, since I usually work on neotropical fireflies, so from the New World, and so I had to actually really do some research for this, and I decided that not just to do Southeast Asia, but also address Southwest Asia, which I guess think India is in. So first of all, let's start off with, so currently there are 12 subfamilies of fireflies in the world. This is a list here, and the last 3 were all from the New World that were described. So we have Chagovariini, Chesperitoini, and Cladoidini were the last 3 that were established, and they're all from the New World. So currently we have just about a little over 150 genera, which includes fossil genera, and over 2,500 species. And I know in most recent papers, older numbers like 2000 or so are usually cited. If you want a good paper to cite for this over 2,500 species, please look at Ferreira et al. 2020. And I will put the link in the chat after my talk where we described Cesperitoini, and there we are actually citing these newer updated numbers. And so in the two areas, so Southwest, Southeast Asia that I'm going to address, you have 4 subfamilies of the 12, the Siphonocerinae, Lampirini, Luciolini, and Odotretini. And then also a bunch of genera that are currently, after Martinet, all 2020, are considered Incertacetes. So some of the genera, we don't really know where to put them, right? So they are currently not classified in any subfamily. So this is basically the region I want to talk about. So anything from Afghanistan down to Indonesia in the southeast there. And so I looked at 18 countries. Sadly, I did not look at China, and Leslie put me on the spot there just a little bit earlier with the species, and I just did actually do a count during like the last talk, and there should be plus minus 107, like there should be 107 species plus minus a few that I may not have looked at the literature yet. So just over 100 species for China. And in these 18 countries that I looked at, the ones in gray, you have 4 subfamilies, like I said, 52 genera were present out of the 150, and 493 species is what I looked at during the preparation of my talk. First, I want to start with the Southeast. That was what I was asked to kind of talk about in the beginning by Dr. Chakravarti. But then I thought it would be nice to include India and some other countries. So you have these countries: Brunei, Cambodia, Indonesia, Laos, Malaysia, Myanmar, Philippines, Singapore, Thailand, Timor-Leste, and Vietnam. And these are the numbers of genera that are currently recorded from any of these countries. So you can see Indonesia has the most genera with 30 known genera. Of all these countries, Timor-Leste is the only one that I could not find a firefly record published. I'm sure they have fireflies. It's just not published currently. And then later on, I will concentrate on the 6 bigger countries. Due to time issues, I couldn't address every country on this list. So I'm going to talk a little more about Indonesia, Vietnam, Malaysia, Myanmar, Philippines, and Thailand. And here is the number of species that are currently present in these countries. And you can see Indonesia by far has the most species currently known with 151. Followed by Vietnam with 70, Malaysia with 62. And then, you know, you have a drop with Philippines 41, 26 in Myanmar and Thailand. And again, Brunei, Cambodia, they have fireflies, but not very big numbers. Singapore actually is funny because they have 6 genera and 7 species. So basically every species is in a different genus right now. And then secondly, I will also talk about this region, which I consider about like the Southwest Asian region. So Afghanistan, Bangladesh, Bhutan, India, Nepal, Pakistan, and Sri Lanka. And I think looking at this was most eye-opening for me because of the discrepancies of knowledge that is present in some of these countries. As you can see here, so India by far has the most genera. So you have 25 genera for India, and then followed by Sri Lanka with 18. But then the other countries like Pakistan, Bhutan, Bangladesh, Afghanistan, so there either are no fireflies or very limited numbers, or that they're really just not studied very well, which given the proximity of Pakistan to India is very surprising that there is only 2 genera known right now. And also only 2 species, and India has 86 known species of fireflies. So I guess I'm not sure if the habitat in Pakistan compared to India is just not compliant for fireflies, or if there's really just a big lack of studying fireflies in Pakistan compared to India. And then Sri Lanka is second on this list with 49 species, which is quite amazing for a small island like Sri Lanka. But I think when we look a little bit into this later, I can show you why that is and what's going on there. So I looked at all the 490 species, 93 species, and I made I was interested in when was this work done, right? So this is a graph that shows species discovery by year, and it's cumulative. So it starts in 1750 when Linnaeus established the binomial nomenclature, and actual like species names were considered valid from 1758 on. And you can see there was little going on early on until basically the 1850s, and this is by decade. So each little field is a decade. And so you can see in 1850, there was a slight incline in descriptions to about 50 species known from, this is for both areas together, Southwest and Southeast. And then really when it starts, it's like the 1880s, 18 to 1930s, which seems to be in fireflies in general, the heyday of firefly descriptions. And this is due to people like Gorham, Peake, Olivier, which all were very interested in fireflies back in the day, and in the British Museum and Paris museums. So you have this really steep incline from 1870s to about the 1950s, with about 450 of those 493 species described in that short area, about 400 species of those described in that short era. And then there is, again, from 1950s to the about 1990s, 2000s, there is a lack of work on fireflies, at least in taxonomy, maybe not in behavior and ecology, but in taxonomy for sure. There was a very calm period until the 2000s, when I guess President Leslie Valentine decided that it's time to clean up the Luciolini of Australia, the Pacific Islands, and the Southeast Asian region. So you can see there's another incline in species descriptions from there on to the current number of about 493. And just to illustrate this a little more, this is species discovery by year, but not cumulative. And here you can see exactly those trends, where in 1850 there is a peak, which would have been Moczulski, basically describing a lot of species, and a couple of other authors. But then in 1880, that's where Gorham started, and Olivier, Ernest Olivier, the second Olivier, his, I think, uncle was working in the 1790s, but he did not really address many Asian fireflies. He was more a New World guy. But in 1880, then Ernest Olivier from Paris, Gorham, who worked on fireflies in Britain, they are like really gotten into it, and they described a lot of species, but it really peaked in the 1920s. But that is when Maurice Peake got into the pictures. He started describing it in about 1911, and then in 1920s he was really productive at that point, I guess. I'm not saying there were good descriptions, but he at least described them. And then you have, like I said, you have that little dip in 1950s, 60s, 70s, 80s, and the 90s, basically. And then in the 2000s, it wraps up again. The 2020s are very low, but we're only 3 years in. I really expect this to go up a lot. And this is for all 4 subfamilies and the Incerticides. So this is a total. So I broke this also up a little bit into the subfamilies. So this is Luciolini, and again, very slow until the 1880s about, and then a big increase, especially Gorham and Olivier did a lot of work. Peake actually wasn't super productive in Luciolini, but he did a little bit. And then again in the 2000s, this is when Christine Lemkin, Van Yuso, Leslie Valentine, all those fantastic researchers from the Southeast Asian region got into fireflies. And Leslie, I think, led an amazing work there in getting all these other fantastic people involved. And we're getting, new papers out every year and really well establishing the Southeast Asian firefly fauna, especially in Luciolini. And here again, by year, not the cumulative. So you can see there's still these two peak periods of descriptions with Gorham, Peak, Olivier, and then Ballantyne et al. Later on. This is for the Ototretini. This is not something Leslie would have worked on, but you can see this is really like nothing happened basically till 1890s. And then in the 1890s until about the 1950s, Maurice Peake was really into Ototretinines, I think, and he described just a whole bunch of them in that short period of time, basically going from 10 species to 140 within 30, 40 years. And then there's a little bit of work done since the 2000s. We have like Brancucci and Geyser from Europe, and then also Janisova and Bokakova from the Czech Republic have been working on some of these Ototretinines. And here you can see it's basically the same trend as in Luciolini, just a little bit, the times are a little bit moved later because Gorham and Olivier did not work on these very much. So it's mainly Peake, and then later on some Europeans again. And I will talk about Ototretinines a little bit more. This is definitely the most neglected group of fireflies in the family. There is an amazing biodiversity, especially in Sri Lanka and some of the other Southeast Asian islands. And there's a lot of work to be done on this subfamily. And then this is for Lampirini and Incerticetus. And you can see this also starts, the peak starts about 1880s, 1870s, and goes up to like 1940s. And then you see there is basically no one has worked on this subfamily since about the 1940s. There's a very few species descriptions for Southwest and Southeast Asia. A lot more people work on this in China, Taiwan, and Japan on these subfamilies, but this is also a subfamily that will need a lot of work, and there's a lot, I think, to discover and figure out for the subfamily and the Incerticetus genera. And here you can see this is very easily to see. You have that huge peak in the 1980s, which, again, Gorham and Olivier, and then a little bit work done after that until the 1940s. Authors. I decided to give you a little overview who worked a lot on this. And these are the spots from 4 to 10. And you can see Leslie is slowly getting up there. She already is in spot number 4 with 33 species descriptions out of the 493. And then you have Walter Whitmer, Bourgeois, Mocholsky, all of them have worked between the 1850s and the 1980s, with Whitmer, I think, publishing until the 80s. And then some new people like Bokakova and Brancucci and Geyser that have described a bunch of Ototretinines in that period. And then Walker, who was basically worked on Indian and Sri Lankan stuff. But who is before then? And you can probably guess it. Gorham described 47 species. Olivier, 107. And then there he is, the king of South Asian fireflies, Maurice Peake. Leslie and I have talked a lot about him. I think we share a dislike of the man. His species descriptions are 2 to 3 lines long, not very informative, and mostly like he compares them to other species that also had short descriptions. So studying fireflies from this region a lot of times does demand you go to Paris, where his specimens are located, to actually look at the specimens and see what he has done. But I would estimate that in world fireflies, Maurice Peak and Ernest Olivier, number 1 and 2, are also number 1 and 2 and have probably described at least 30 to 40% of the firefly fauna of the world. So now I'm going to go a little bit into the countries. So I'm going to start with the largest, or with the country that has the most fireflies in Asia, in Southeast Asia. Indonesia has 3 of the 4 subfamilies and some of the inserter cities. Like I said earlier, we have 30 genera, 151 species, which is quite amazing. But also, I'm not very surprised. So I studied the West Indies, and I can totally see that island biogeography lends its hands to a wide and big species biodiversity in a lot of things. And fireflies seem to be very much thriving on islands. As in the West Indies, Hispaniola, the island that has the Dominican Republic and Haiti, harbors about 200 species, which it's not a very big island. And it's quite amazing that such biodiversity can establish itself there. And so I'm not very surprised about Indonesia having these many species. And again, with islands, 76 of those 151% of these 151 species seem to be endemic to Indonesia and those islands. And I again made a graph. This is showing these species description curves for the islands. In blue, you have per year, and in black, you have cumulative. So you can see that we have the same trend between the 1980s and the 1940s is where the majority of species descriptions happened. And then you can see again, it's definitely climbing since the 2000s. And this is based on Leslie Ballantyne's and Juan Yuso and some of these very great other workers that are studying fireflies now in these areas. Next is Vietnam. Vietnam has all four subfamilies. So this is the only.
Now the speaker has disconnected. Ladies and gentlemen, please wait for a minute or two. He is reconnecting.
I'm very sorry. My computer decided to turn off based on its battery. I will share again. All right, everyone can see this?
Yes, your slides are visible. Yeah, please go ahead.
So Vietnam actually has all the four subfamilies, which is, I think, the only country that might have. So it has several, it has all the four subfamilies, and it has 25 genera currently known, 70 species, and 71% of those are endemic. This, I think, is seems to be a very normal trend in at least Southeast Asia. And here's again the species description curves. blue per year, black cumulative. Again, same trend, 1880s to 1930s, 40s. And then a little bit work has been done since 2000. Again, this all comes down basically to Luciolini being studied, all these new genera being described that, when you look at McDermott 66, there were what, 5 genera of Luciolini. Basically everything was a Luciola. Now there are what, 31 species, genera of Luciolini. So a lot of work has been done. Malaysia, 3 subfamilies, 20 genera, 62 species, and 71% of those are endemic, which is kind of surprising, I guess, a little bit, because it shares so much border with Indonesia. So you would think there would be a big overlap of some of those species with Indonesia, but it seems like not, which may just be a sampling or study bias right now, and we will find out that a lot of these might be overlapping actually. And I think a lot of this endemism comes from Odotretini, which have not been studied in Southeast Asia very much lately. Here you have the curve, basically the same, but look at those really deep, steep incline in species from 2010 to the 2020s, which I think Van Yuso and some of her co-workers and Leslie are responsible for, but Malaysia seems to have gotten the most attention of all these countries lately. Philippines, another island country, state. So we have 3 subfamilies, 13 genera, 41 species, 76% endemic. I honestly expect a sharp increase in species from Philippines soon, when I think some more attention is placed on that region and people will study fireflies a little more. I think we will find that there is at least double that. Here, same, the biggest incline from 1880 to 1890, I think, which probably had to do with Gorham. He had much material to look at in London. And then there's already some work going on in the Philippines lately, in the 2010s. Then we have Myanmar, 3 subfamilies, 13 genera, 26 species. Given the diversity in Vietnam, I think after Myanmar receives some studying, some attention, we'll see that go up a lot. 54% endemic. This is because I think it shares the border with India and some of these other countries, Laos, Thailand, Vietnam. So there is quite the overlap. Here, same trend, 1890s, and recently the species went up. And then Thailand, interestingly, 3 subspecies, 12 genera, 26 species, 38% endemic. Again, it shares the borders with all Malaysia, with Laos, again, Vietnam. So here I'm not surprised that they don't have a lot of endemism. But maybe it's because they haven't studied the species that much there yet. And here you can see this, maybe of all the countries, may have received more attention recent years than it has in the 1800s, to be honest. There was a good amount of species described in like both periods, about half and half. So I think Thailand is on a good way of getting to know their fireflies a little better. Then here, India, 3 subfamilies. Oh, we're switching now to Southwest. India, of course, 3 subfamilies, 25 genera, and 84, maybe 86 species. 2 of them I'm still trying to work out. They are questionable if they are in India or not. I think both are recorded from Indonesia and from India. So that would be quite the range without having any of the countries in between. So that's something, I mean, someone maybe have to look into, are those species really present in India? Because I think they have been established in Indonesia, but records show they could be in India. India has 58% endemic species. Very interesting. It's a big country and a lot of borders, so there's a lot of shared species. Here you can see most everything has been done in the 1850s, but also some work has been done lately, and this is, I think, mostly with again Leslie, and then also some Ototretinine. People have described species from India, but you can see not very many, only like 9 species or so described in the last 20, 30 years. Sri Lanka. I think you heard a talk about Sri Lanka before today or yesterday. I have 3 subfamilies, 18 genera and 49 species. I guess I did not go for the endemism, but the endemism is very high, I think over 80%. And here you can see most of the species were described from 1860 to 1970. Peake and Whitmer were the most prolific people here, and then a little bit is going on lately with Dr. Wisha Kuhn, I think, and some of his students been working on, here's the endemic part, 73%. And then last, I addressed Nepal. The other countries didn't have quite the diversity. 4 subfamilies, 10 genera, 18 species, and this is mainly Ototretini that the diversity comes from. And here you can see actually that there were some descriptions in the 1840s, a little bit in the until 1970, but the majority of species have been described since about 2000. And this comes from papers by Brancucci and Geiser, and then also Janiskova and Bokakova from the Czech Republic. Those were working on this material and described quite a few species since. 67% of the species are endemic. And then here, I know I've been talking about Leslie, and this is just a small little cut of the papers she has done. And I'm sure if you contact her or me, and you want to have some of her papers, I have all of them, of course. You can see she's very prolific, and she's been publishing a paper or 2 every year. And with her big group, Dr. Fu from China, Christine Lemkin from Australia, Ming Lian Zheng from Taiwan, Dr. Wichakun from Sri Lanka, Van Yuso, Anshara Thancharon, and I mean, all these people. I'm sorry, I'm probably forgetting a lot of people here that she's worked with, but it's quite the prolific group of people that have paid so much attention to the Luciolini of Asia. And we can only hope that Leslie is around long enough that she can tackle the Luciolini of Africa, because who else is going to do it, right? And so my conclusions are, a lot of work remains, and that is the case for every region of the world. We have a lot of work to do everywhere, but this is about your guys' region, so much work remains. Maybe not as much in the Southeast Asia region because it's on a good trajectory, but then again, Ototretinines and Lampirini haven't been touched in a long while, so hopefully people start looking into those subfamilies and insert cities. The Southwest Asian region with India and Pakistan, you guys have such great potential for us to figure out what's really going on and how many species there are. And I hope that maybe someone from Pakistan is on this call and watches this and is like, oh, we have to look into this, like 2 species. Doesn't seem right. I don't know. And I urge everyone to use good taxonomic practices. Compare your specimens with known and authoritatively identified specimens. Compare them to the type material, which I know means sometimes having to travel to Paris or to London. Or so, but it's good practice, and you will, not have misidentifications and, and you will find that there is a lot of synonymies. And like when you look at Leslie's work, a lot of species that Pic, Olivier, and Goyen described, they were synonyms of other valid species. And so all these things have to be looked at. And look at Leslie's papers, looked at Juan Yuso's papers that have been published recently, and take those as an example for really good taxonomic work and how a revision of a certain area or a certain genus or group of species should be done when it's published. So we have quality work coming out of every place, and it's the same in the New World. We have a lot of people working on it, and we want it to be done right. So I think you can contact Leslie. I'm sure she will help you in getting you started. You can contact Ishwari. He works on Mexican fireflies. You can Veronica, Juan Yuso, and all those people that have done a lot of work on taxonomy lately. Contact those guys and get some advice, or you can contact me. I have the world's largest library of PDFs on fireflies. I have over 3,500 firefly PDFs spanning the whole world. Have every single species description ever published in my library. And I am, very close to be done with a world checklist that hopefully will be submitted soon, that has all the 2,500 plus species and 151 genera addressed. And for the first time also we'll have a bibliography of all the species descriptions, all the papers, which, I have to pay a lot of admiration to Olivier and McDermott, who produced the last lists. Given the times they lived in, to produce a list with 1,200/1,900 species without the internet, without, with having to write letters to people to get all this literature, was an amazing task. And I really admire what they did. And so I hope that my list can follow that up and help people, in their works. And then I urge every one of you guys work on odotretinines. This is a group that needs a lot of work. There's a lot of amazing species, like you can see on the right side. This is Lamellipalpus constantini. Look at those palps. I mean, they're just, every single species is just amazing in this subfamily. We only have two members of this subfamily in the New World in the US. Every single other, of these guys is in Asia. And I hope, people put as much attention to this group as they did, as Leslie and Co did to the Lucy Alliance. And with that, I would like to thank Leslie. We had many discussions about fireflies in the past, 10 years, 9 years now that I know her. Dr. Chakravarti and EMPRI for inviting me to give this talk. Ainsley Siago for producing this amazing graphic for my talk. And then FDAX, the Florida Department of Agriculture, Department of Plant Industries, and the Florida State Collection of Arthropods for helping me and supporting my firefly work. And with that, I will take questions.
Thank you, Dr. Oliver Keller, for a very important updated overview on the distribution, occurrence, and the taxonomic revisions the Firefly group has undergone, especially in Southeast and Southwest Asia. We fully agree with you that there is a great potential for us to work in Southwest Asia, and we will be looking forward to your collaboration, help, assistance in developing the regional checklists, especially country-wise. So as far as India is concerned, we are at the preliminary stage of preparing checklist for India, and we have more than 86 species. So you have listed about 84 or 86 species, and as per our what you call preparation, we have more than that. Anyway, we would share with you, and we would like to update that checklist with all the details and descriptions. We have two questions for you, Professor Oliver Keller. One is that one of the participants has asked a question. Does diverse geography or geographical conditions contribute to species diversity in fireflies?
I would say yes. It's a hard question, I guess. So fireflies are not very good flyers, like they don't disperse very much. So if you have a landscape or a biogeography that has a lot of different mountain ranges, for instance, you will have a lot of new diversity coming up because they don't disperse very well. So my experience is from Hispaniola. It's a very small island, but it has 4 mountain ranges that all transverse the island. And you find new species in between all these mountain ranges. And a lot of these species do not share the habitat, nor do they occur on the other side of the mountain range. And there's only like less than 5% of the species are more like distributed across the island, while most other species, some of the species you can only find in one valley of the mountain and nowhere else. So this also goes to where, you know, you have to very carefully look at the species, because you might have a different species than you think in a different area just because of geographic barriers. And you might have to dissect the species. You know, the flash patterns may be very similar, but somewhat different. But, you know, a dissection of the genitalia might show you that they're different, but it also might show you that they're the same. So this is why, yeah, you have to really, like, you know, get familiar with the biogeography of your area. And, you know, you have India, you have a lot of very, you know, you have the coastal plains, you have the mountainous regions in the north. So I would assume, yeah, you have a lot of biogeographic, very different areas that would lend itself to a very different kind of yeah, firefly. Like, you know, you look at Sri Lanka, you have all these Ototretna, and Sri Lanka is an island. So in your coastal areas, I would expect that you will have a lot of Ototretnines as well that may have not been studied very well. And Ototretnines do not produce light very often, right? There's only a few that do. So you would have to go and look in, you know, you do a sweep netting during the day to find these kinds of species. But yeah, so, and for what you said about your list of species, so I am very much limited to the literature that is available to me. So, you know, I might not have literature that you have. I might not have literature that's written in Indian or, you know, in your language that I would never find in my Google searches, you know. So if you, anybody that's on this seminar, if you have literature that's written in your native language, and if you would share that with me, I would really appreciate that, because this is something that I have a hard time access sometimes. Because it doesn't show up, or I don't have the ability to get the literature based on my library not being able to get it for me. But I would really appreciate that, because the checklist I want to produce, I want to be as comprehensive as possible and give the best overview we can about where our fireflies located. And my numbers are from what I have accessible. I am very well aware that for most countries, I'm probably going to be off a few species because of lack of literature and information, yes.
It's very true, and it's very factual, Dr. Oliver. Now, one more participant is asking, without handling the firefly specimens and without studying them under the microscope, can we identify firefly specimens just by photos, by having photography?
To genus, I would say yes, that's very much possible. To species, even to genus, it's sometimes you will need more than a ventral view, because, you know, I love how many people are into a naturalist and observing fireflies in India. I'm in a WhatsApp group about Indian fireflies, and I see all these pictures. But the problem is the view from the top is not always helpful in identification. Oftentimes you will need a view from the bottom, like the light organs. But even then, species identification is very hard in regions that have 50+ fireflies. I mean, I live in the US. I cannot identify every American firefly species based on a picture. It's just not possible. I can give you a genus in 90% of the time. I cannot give you a species most of those times. So it's very helpful to have all these data points all over the world, given iNaturalist, given all these other aggregators. But for a positive species ID, you will, in most cases, need a dissection of the specimen, of a male specimen. If you have a female, identifications are even harder, sadly. Leslie has done a good work with a bunch of females being pictured and imaged or being described. So you can sometimes identify them, but the general gist, I think, for the world is females are a lot of times unknown. Identification relies on males, and you will need genitalia.
Yeah, that's very true. Any more questions from participants to Dr. Oliver? Yeah. If there are no questions, then we handsomely thank Dr. Oliver Keller for devoting so much of his precious time with us and telling us very important and basic information about fireflies. And definitely he needs our help and our coordination so that the world checklist and the world of fireflies database is enriched. Sir, we are at it, and definitely we are under the guidance of Professor Leslie Valentine. She has been helping us in all our activities, and we hope that we will be able to update the checklist from India. The another most important event, forthcoming event that we would like to touch upon is the taxonomic workshop. This will be very useful for the beginners from all over the globe, and we are working with IUCN, Professor Leslie Valentine, and also with Dr. Vaughn Joshua in organizing a taxonomic workshop for the beginners so that it becomes a hands-on training and come out with a small manual so that at least the local species of fireflies are identified, and we have good capacity building in the generations to come. Thank you so much once again, Dr. Oliver Keller, and definitely we'll be in touch with you. With this, we end the 2-day webinar on fireflies, and we handsomely thank all the speakers, their institutions, team, NGOs, universities, to have devoted and shared their valuable time and experience with us. It has the response has been very overwhelming, and the talks were highly illuminating, and we could see some of the trends, techniques, approaches which have been applied in understanding firefly research and take firefly science forward. We are also indebted to the IUCN for their support in all possible ways, the Entomological Society of America for putting this event on their platform, the Department of Entomology, University of Sciences, Bangalore, GKVK, and the Naturalist School, who have helped us in organizing this webinar at a very short period of time. So with all the things at place, we also thank the participants because we will be able to know that people who are interested in firefly research, science, hobby, ecotourism, we all are very much essential to see that in the times to come, fireflies are effectively conserved, understood, and appreciated. So with these few words, we again thank each one of you who have contributed to this webinar, and especially our thanks and gratefulness goes to Professor Leslie Ballantyne, who has been with us throughout from the first minute to the last minute. Madam, we thank you woefully for this, your presence and your comments and sharing your valuable experience and all the helping us in all possible ways to understand fireflies better. Thank you so much. Yes, thank you one and all. Hope to be in touch with you all. Good day. And we have one feedback form. You can fill it up, and definitely we will be able to in touch with you, and we will catch up with you for a workshop on taxonomy of fireflies as early as possible. Thank you once again to each one of you. Good day.